Hi, everyone. We're just going to give it a few minutes for people to get dialed in. Hi, Vincent.
I'm guessing I'm in the right place. I'm in the panelist part.
Yes.
Okay.
Uh-huh.
I made it.
Perfect. Yeah, we're just going to give it another minute or two for people to get dialed in, and then we'll get started.
Great.
Connie, I control the slides from through the program, right?
Yeah, I'll give you permission to present as I'm finishing up, or maybe Michelle could do it as I'm finishing up my slides, and then you should be able to switch over, and then I have yours as a backup.
Thank you.
Great.
So is that the case for all of us? I'm driving the PowerPoint on my slides or through the program?
You can. I just have it as a backup.
Okay. Okay.
Okay, well, we have a few minutes past. I think in the interest of time, we can go ahead and get started. So I'm going to kick off the meeting today with my slides. And panelists, feel free to have your cameras on when you're speaking, but I ask when you're not to just turn it off. And so, great. Hi, everyone. Welcome to the Botanical Safety Consortium annual meeting. This is our second annual meeting held virtually again. I'm Connie Mitchell. Along with Dr. Michelle Embry, I co-manage the consortium. I'm a scientific program manager from the Health and Environmental Sciences Institute, or HESI. I'm going to kick off the meeting today by going over some logistics and objectives. And here is our agenda today, and it's also attached as a handout on GoToWebinar if you want to view it. Give me 1 second. We have a 4-hour meeting scheduled today, and after I'm done speaking, Cara Welch is going to kick us off with some background information on the BSc. And just for everyone's knowledge, the meeting today is going to be recorded and it'll be available on our website in a few days. Each talk will have a few minutes at the end for clarifying questions, and then we'll have an open discussion at the end of the meeting. So after Cara gives her talk, we're going to have the first of our 2 keynote talks from Dr. Grienke from the University of Vienna. After that, Dr. Vincent Sica is going to give a talk on the chemical analysis work he has done on one of the botanicals we're piloting, Ashwagandha. Then Dr. Lawless is going to present modeling work with botanical constituents he and other consortium members have been working on. After a short break, Dr. Sutherland will talk about the new approach methodologies we are initially piloting for botanicals as complex mixtures. And then we'll have our second keynote from Dr. Lodog, who will share some of the challenges facing clinicians with respect to botanical safety. We'll have a few concluding comments and then time for questions at the end. And here's our objectives for the meeting today. So we want to provide an overview of the BSc for folks that are new or not as in tune with what we're doing. And we also want to highlight our progress over the past years. And our keynote speakers today will highlight ongoing work in the botanical space. And we're also looking for new stakeholders to give us input and add their expertise, in addition to soliciting input from our current members. And this slide shows the logistics for the meeting today. So like I said, the entire meeting will be recorded and we'll put it on our website within a few days. And we'll also share the PDF slides for the talks with permissions from the speakers. And everyone will be muted other than the panelist, but please throughout the day, feel free to ask questions in the question box. And I have a slide on that on the next slide. And after each talk, we'll have about 5 minutes for clarifying questions along with some time at the end of the meeting. And during the meeting, if you have any issues, you can email Dr. Michelle Embry or myself. All right? So this shows how you can ask questions. So if you're on the desktop on the left, you can enter it into that box, or on the web instant join, there's a little question mark. And just before we get started today, I wanted to give some brief information on HESI, my organization, which is the convener of the Botanical Safety Consortium. So we are the Health and Environmental Sciences Institute, a science not-for-profit that has been operating for over 30 years in Washington, D.C. And we are working towards improved safety and innovation for human health and environmental health by bringing together experts from industry, academia, government, and other non-government organizations. And this slide shows our focal areas, which the BSC fits into multiple, along with the other committees we have. And for the BSC, we're leveraging expertise from all, but I bolded the ones that are most relevant for the Botanical Safety Consortium. Then on the right are our project mechanisms that we work through, such as creating decision frameworks, generating novel data and experiments, writing open access manuscripts, and holding scientific meetings and trainings. And for more info on the other committees, you can go to our website, hesiglobal.org. And so with that, we can dive into the meeting. I really thank you all for being here today. And I thank all our current members that have worked so hard to move the project forward this last year. And thank everyone for tuning in today to hear about us and our updates. And there's just our contact info for the Botanical Safety Consortium website, along with my email and Michelle's. And now I have the great pleasure of introducing Dr. Cara Welch. Cara is the acting director of the Office of Dietary Supplements Program at the US FDA. And we also have the honor of having her as the FDA chair of the Botanical Safety Consortium Steering Committee. Thank you so much, Cara. I'll turn it over to you. Let me give you access real quick. Okay, you're now a presenter, so you should be able to share your slides.
Wonderful, thank you. Okay, so good morning, everyone. I am so pleased to be here with you all today. Whoops, I just lost my slides. There we go, sorry. So pleased to be here with you all today. My name is Cara Welch, and as Connie said, I am the acting director of the Office of Dietary Supplement Programs at the Food and Drug Administration. I am, as she also said, I am here more in my capacity as a member of the steering committee for the BSC, and I have the distinct pleasure of introducing the BSC for anyone who is new to the consortium. Maybe you haven't been involved with us over the last year and a half, as well as providing a brief update on what we've been doing over the last year. It's been an exciting year for the BSC. We've made a lot of great progress, and I think a slide later on really demonstrates sort of the development that we've had since last year's annual meeting, and the input we received during last year's annual meeting was really helpful as we continue to sort of build out the BSC and our strategy for the upcoming year. So getting started, sort of talking about botanicals. Everyone on this call is well aware of botanicals and sort of the complexity that they bring. Botanicals are factories of bioactive chemicals, and the breadth of medicinal plants is so wide. And I know we're going to hear later on from Dr. Lodog, but the Kew Botanic Gardens discussed in 2017. Close to 30,000 plant species have been recorded as being used for medicinal purposes. And we've really just sort of scratched the surface of that. I think a fraction of them, of these 30,000 plant species, have been cited in medicinal regulatory publications, sort of digging into, do we understand the botanical and the activity of that botanical? And so there's so much work to be done, really. looking at complex botanical ingredients, they're full of, bioactive constituents, secondary metabolites. And I think it's sort of within our nature to dig into these botanicals, find the bioactive constituent, maybe the most active constituent or the most stable and active constituent, and sort of turn one active constituent into a pharmaceutical or a pesticide, maybe a cosmetic ingredient. The BSc is actually taking a different approach, and what we want to do is really sort of facilitate the use of botanical ingredients as complex botanical ingredients. Botanical ingredients have experienced a really healthy growth over the last 20 years or so. The figure on the left side of your screen shows just U.S. retail sales of herbal supplements, not even other consumer products. In 2017, it was about $8 billion. Over the next couple of years, shot well over $9 billion. So really expansive growth. And I think this growth is largely coming from, looking at the use of botanicals in disease management, the use of botanicals, sort of integrating them into the healthcare system. And this has developed or has led to, real continued expansion of botanicals. And then, of course, over the last year and a half during COVID times, we've certainly all recognized the increased sales for any botanical products with immune health properties. And I think the goal is not just to demonstrate, the increased usage of botanicals, but the goal is to make sure we can use them safely. And the botanicals present some challenges on that end. I think the toxicity testing, the overall safety evaluation processes for consumer products are often built around and optimized for single chemicals. If you're looking at a drug or a pesticide, generally we're looking at a single active pharmaceutical ingredient, whether or not it came from a botanical. These products, the end products for drugs and pesticides are simple, they're consistent, right? Because they're just a single chemical entity, maybe 3 active ingredients mixed with some inactives. The biological activity is really closely associated with the active constituent. Botanicals are a different beast in almost every one of those areas. So botanicals, we have complex ingredients. They're variable, and there's a lot of opportunities for variation in botanical ingredients. The biological activity is often associated with the entire mixture. We don't always know what the source of the activity is, or, as I suspect in many cases, the sources of the activity, right? Because constituents work with each other. Maybe there's even some mitigating effects when you're talking about a complex botanical ingredient compared to a single constituent or class of constituents. And as I mentioned, there's so many opportunities for variation in a botanical ingredient. Starting off at the raw plant material stage, the biomass that you're looking at can differ based on the weather conditions and the soil conditions, the season of the plant and the maturity of the plant, all sort of adjust the chemical makeup of the particular plant material. Carrying that over to processing the botanical ingredients, the extraction process and extraction solvent is so important to figuring out what constituents you're pulling out of that plant, that biomass, what the safety of that material will be. Carrying that into the finished product, of course, now we have a complex botanical ingredient being mixed with other ingredients, inactive excipient type ingredients, combining with other complex botanical ingredients. How those ingredients sort of play off each other also obviously affects the safety of the end product. And then carrying it all the way to sort of a post-market evaluation of the safety of the product, we have to take into account the human element, the actual consumer taking the product, their pre-existing conditions or age, genetic makeup, as well as, how was the product consumed? Was it within the described conditions of use? And I think understanding all of these different factors it really presents a really complex question as to how can we make sure we understand the safety of the end product at the various stages from raw plant material all the way to the end product. And so, we often have problems then, and I don't mean to say that botanicals are problematic necessarily, but they do present their own specific problems. We have limited botanical safety assessment framework or approach for botanicals. Often we rely on history of use, decades or centuries of use as a food, and how that can be read into the plant material, the botanical ingredient, as well as the finished product. We often rely on in vivo testing, and animal testing, of course, creates its own challenges. It's costly and timely, or I should say time intensive. There are some ethical concerns with animal testing. And so I think, the goal would be, can we move away from relying on history of use in animal testing? Also, of course, looking around the globe, the safety requirements and what comprises sufficient safety substantiation changes from country to country, and the different type of regulatory systems that you are utilizing to introduce your product. And then, of course, I think ultimately when you're looking at the formulated products on the market, I think it's safe to say we generally have little or even no toxicity data on some of these formulated products. We may very well have toxicity data on specific constituents or a class of constituents, but sort of reminding back to the sources of variation, ensuring how that information can be read into a complex ingredient or even a product that contains multiple complex botanical ingredients, it can be really difficult. And so, a group of us, and I use us loosely because I was not necessarily part of the initial conversations, but a group of us were sort of sitting back and saying, there's all these problems with botanical ingredients and the safety assessment for botanical ingredients, and what can we do about that? So we developed the Botanical Safety Consortium. The BSC actually started really organically with some conversations, conferences, workshops, meetings of the mind, and kicked off with a publication documenting a roundtable discussion in early 2019, talking exactly about those problems and our idea for an approach. FDA publicly announced our support for the BSC in early 2019 as well, and specifically how the importance of this work, the work of the BSC, can advance FDA's regulation of botanical dietary supplements. The goal was sort of tied into our broader modernization, and this partnership of industry and academia and government can really sort of tie it together and give us a good perspective of how to evaluate the safety of botanical ingredients. So as Connie mentioned, the BSC was officially convened with HESI in late 2019. A memorandum of understanding was signed with FDA, NIEHS, National Institute of Environmental and Health Sciences, and HESI. And that officially convened the BSC. It sort of kicked off our work in early 2020, and then of course our first annual meeting, I think it was May of last year. And sort of looking at the BSC, I think we can all sort of understand this problem statement that I introduced at the beginning, right? Understanding, do we know which approach methodologies can be used when we're evaluating botanical safety? all the various approach methodologies and how they can come together to ultimately provide us where we want to be, a strategic framework and toolkit to evaluate the safety of complex botanical ingredients. The objectives of the BSc are on the screen. We are looking to engage with a broad group of stakeholders, of global stakeholders, many of which, of course, are in the audience today. And that is really important because we want to ensure that we have the best approaches at hand. We understand, which approaches are sort of foremost in the scientific field, but also feasible for regular use, right? So because of that, we need industry and academia and regulators at the table. We're looking to establish an appropriate level of chemical characterization for these complex botanical ingredients. I think any one of us, and in fact, many of us sort of took a particular botanical and did a sort of dissertation style research into a specific botanical, and we can categorize the classes of constituents and the new chemicals in a particular botanical. That's not the goal here. I think the goal here is really to sort of get an appropriate level of chemical characterization so that we understand what the botanical is, how what we are looking at relates to what was traditionally used in the publications, but also what is being marketed, and understanding how they relate. We want to identify a pragmatic and fit for purpose assays, specifically in vitro and in silico assays to evaluate botanicals. We will actually be applying these assays to identified botanicals and getting all of that information, and then ideally taking all of these results and sort of integrating them into a toolkit and a framework for a more holistic viewpoint of how to evaluate the safety of botanical ingredients. So those were sort of the broad objectives of the BSc. The current charge of the BSc is to evaluate the suitability of assays for complex mixture, for botanical complex mixture. What we are not doing is, we are not looking to develop a determination of safe and unsafe ingredients. That's not the goal. We are not making regulatory decisions. FDA has been part of the BSc from the beginning, and we find it so important to be part of the conversation. But the goal is not to then take the BSc output and make it law for FDA, right? That is not the goal. The goal is for that to be a toolkit for industry to use, as well as for FDA to use. We're not endorsing any particular product or method. We are not testing finished products. We're not testing individual constituents other than positive and negative controls. We are testing complex botanical ingredients, extracts, crude extracts. At this point, we are only looking at oral routes of administration. We talked a lot last year's annual meeting about expanding into topical. And I think we all accept that that will be where we go. But right now we are focused on oral routes of administration. We are not performing animal testing. We are not performing risk assessments, and we are not the big V validating method. We are evaluating the applicability of assays, but we're not doing validation. So I think this is probably a good slide to demonstrate the progress over the last year. I think we talked about some of our technical working groups at last year's annual meeting. And because of the discussion during the annual meeting, we ended up forming even more technical working groups. And so this slide is a good picture of all of our technical working groups. Sort of reading actually from right to left. And in the very foundational sense, we have a lit review work group. And the literature review group really digs into the literature and makes sure we have a comprehensive picture of what the literature has for a particular botanical, for example. The analytical work groups, we have a chemical analysis and data analysis group, as well as an IV-IVE group to make sure we can sort of make that jump, that extrapolation from in vitro to in vivo. For exposure, we have ADME and a botanical drug interaction work group. The toxicity endpoints, and I know we certainly spent time on that last year, but we have technical work groups, specifically for hepatotox, cardiotox, neurotox, genotox, DART, and systemic tox. And those groups are very active right now. And then sort of across all of these themes, we have a pharmacognosy work group. And they are in a bit more of an advisory role, and their advice weighs into so many of these different stages. And so I think this is another new slide, a good picture of the development of our work over the last year, a detailed look at our strategy, starting from sort of the questions, the priority endpoints, how do we test for hepatotoxin, cardiotoxin, genotox, all the way through to the end of taking all of this data and integrating it into a toolkit and a framework to evaluate botanical safety. So we are starting off on the left side of the screen with our endpoint technical working groups and asking them to sort of develop lists for us of botanical candidates as well as assays, to identify assays and approaches to analyze botanicals. So, digging into the literature, the priority endpoint work groups are identifying botanical candidates that hit each of their own specific technical working group, their own specific endpoint, as well as identifying assays, in silico and in vitro assays that fit their endpoints. We carry this over to the analysis work groups, which then can provide us sort of a compiled library, both a compiled library of botanical ingredients as well as compiled list of assays. We will then apply the assays to the botanicals, analyze this massive amount of information, and ultimately turn it into a testing framework, an evaluation framework. The next couple slides are actually going to focus on a lot of the work we've done over the last year, sort of on the left side of the screen where the pathways split. So when we ask the endpoint work groups to focus on, to identify assays for us, we ask them specifically to identify in silico methods and screening assays, but also to identify mechanistic assays for mechanistic follow-up. And that's really important there. So making sure we understand sort of the initial look and the screening of the botanicals, but can we understand the mechanism of the activity? We also ask them to focus in on the specific guidelines for which assays they chose. We wanted them to identify assays that were established and reproducible, accessible, very important. We want to make sure the framework is accessible for use outside of just sort of the best of the best. But what do we have here that are suitable for botanicals, accessible for use by many groups, reasonable cost, but also, of course, relevant to their mechanism of action to make sure we can evaluate the toxicity. And then also the list of botanical candidates. And this was really important work, of course. So we asked the technical working groups to identify botanicals that hit on their specific endpoint, based on in vitro evidence, in vivo evidence, clinical evidence, maybe case their tox endpoint. And what each of those 7 groups put forward was a long list of botanical candidates. In some cases, some overlapping, in other cases, not overlapping. And we took that long list of botanical candidates and fed it over to the next group of work groups. So starting actually from the bottom up, the pharmacognosy work group provides some, again, sort of really valuable information. They are helpful to ensure we understand sort of the historical use of the botanical, what was it indicated for, what types of dosing do we see the efficacy at, what types of dosing do we see the adverse effects, but also, you know, identifying a source or a supplier of the material, because we do actually need to test the material. The literature review work group did a lot of work to sort of gather information and provide us a comprehensive picture of the botanical candidates, again, for both their uses and their toxicity information. And the chemical analysis group provides some information to ensure that we can identify the botanicals. Can we authenticate the material? Are there marker compounds that we can use? Both for identification and quantification, you know, what's the stability of the material? And so all of that input sort of took a long list of botanical candidates and narrowed it down to a more reasonable number. And then there was some additional discussion, you know, ensuring the material, you know, feasibility to work with, labs that can work with it. And we ended up with a list of botanical candidates that I'm actually going to go through, I think, next. Yeah. So the final step was sort of taking a list, 20, 30 ingredient botanicals, and narrowing it down to these 13 case studies. And to be clear, these are our initial case studies. So these are the 13 that we're starting with. And we're already starting to access material and, you know, get it to the chemical analysis work group to start that chemical characterization. As you look through this list, and I am going to go through them over the next few slides, but, you know, some of these ingredients that you're seeing here are known toxic botanicals, in some cases illegal to be used in various jurisdictions around the world. Others might be a little confusing as to why they're on here. Asian ginseng and milk thistle are actually on here as our negative controls. So they are, you know, demonstrated to be safely used. And that's important, right? As we are evaluating our assays, we need to know, we need to have botanicals that sort of confirm a hit for a particular toxic endpoint. We also need to have botanicals that sort of confirm a negative for a particular endpoint. And finally, we need to have some ingredients that maybe challenge that and help us sort of gain information. So some of the ingredients listed on here don't really fall into either category safely. They're not necessarily considered to be, you know, widely considered to be unsafe. They're not widely considered to be safe. There might be some demonstration of hepatotoxic concern or cardiotoxic concern. You know, maybe the literature isn't quite clear yet how it should be used. And that's important, again, right? We need to be able to challenge the assays that we're working with. I'm actually going to go a touch out of order here. So first talking about the 2 negative controls, Asian ginseng and milk thistle. Both of these ingredients have a robust history of safe use. Both of them have animal tox information as well as human clinical information demonstrating safe use. And so these are our 2 negative controls. And then starting off with some of the positives. So Aristolochia is a great example of a botanical that has known toxic constituents. So the aristolochic acid, we have human case reports and clinical studies demonstrating kidney damage and bladder cancer, has led to restricted use in many jurisdictions. Ashwagandha, and ashwagandha I think is another one of those examples of both there is demonstration, there's published information demonstrating safe use of ashwagandha, but also case reports describing hepatotox and cardiotox concerns. So it'd be interesting to see where the data comes out on this. Blue cohosh. Blue cohosh has demonstrated some concerns with pregnancy. So we have demonstration not recommended for use in the first trimester due to cardioactivity, embryotoxic concerns. Comfrey. I think comfrey is another one of those examples of known toxic constituents, contains the pyrrolizidine alkaloids, which have published reports of demonstrating hepatotox concerns and carcinogenicity concerns. Ephedra, another example of something that has illegal or restricted or banned use in many jurisdictions. So ephedra contains the ephedrine alkaloids, which can demonstrate cardiovascular and CNS effects, as well as cerebral effects. So we'll see where the data comes on that. Green tea extract. We've definitely heard some concern with using green tea extract. I think this is another great example of something that has centuries of use demonstrating safe use, or at least of green tea. But to be clear, what we're looking at here, of course, is a green tea extract, and particularly following up on some of the published case reports for hepatotox with highly concentrated green tea extract, as well as botanical drug interactions. Golden seal. Golden seal is another one that has some contraindications for use in pregnancy, as well as potential gene tox concerns, carcinogenicity. Kava root. The kava root is another example here where it has reports of safe use, but also reports of kava-related liver disease. And kava root is another interesting botanical. The cultivar can greatly change the chemical makeup. Right, there's a lot of sources for variation. So ensuring we have the chemical characterization of the material we're working with and how we can translate that to any results. Kratom. Kratom is another example of restricted or illegal use, illegal for use in certain jurisdictions, less restricted in others. Not sure that we're actually following up on the psychoactive preparations, but more on the hepatotox reports associated with kratom consumption. Usnea, and I think this is another example of, has a history of use, certainly, but we also have signals for reproductive toxicity, particularly associated with an NTP study. And then the last one here, yohimbine and yohimbine specifically causing dilation of blood vessels, potential carcinogenicity as well. And so that is the last of our case studies. So at this point, I am happy to turn it back over to questions. Connie, I'm not sure if I turn it back to you, but I am so pleased to have a lot of our steering committee members on the call as well to handle some of the questions. So thank you.
Great, Cara, that was fantastic. We did have a couple questions come in through the chat. So I'll ask them to you, and like you said, other people as panelists can chime in too with thoughts. So the first is, are toxicological endpoints used for botanical selection from direct primary effects or indirect secondary mode of action? Okay, I can't hear you anymore. I don't know if others can.
Sorry,
there we
double
go.
mute. I was just saying that that is a good question, and I would definitely look to some of my steering committee members who are more involved in the technical working groups as to what went into the conversations as they were developing their list of candidates.
Yeah, I can answer a little as I sit in on a lot of the calls. So I think as you went through some of the slides, a lot are maybe the indirect, what we see in either people or in whole animals, versus some, especially say for genotox, we can see the direct effects, maybe on DNA mutation. Okay, anyone else want to chime in real quick on that? Okay, a few more questions are coming in, so I'm going to go ahead and move on. Curious why botanicals with well-established food use with known safety, such as onions or spinach, were not included as negative controls.
Yeah, ultimately, just checking, make sure I'm not on double mute again. Ultimately, I think it was demonstrating we have to make some cuts, and so I think those are additional excellent examples. But we decided at sort of our initial round of case studies to look at, again, it was the Asian ginseng and the milk thistle. I don't know that there's particularly a good or better choices. But, the two that you just listed are great examples as well.
Great. Okay. So another question that came in is the plan to select a single exemplar of each botanical for study or to compare multiple products?
And again, Connie, I'm not sure if you're better positioned to answer this question. It's a little awkward to then ask and answer it, but.
Yeah, I can. And the plan is, so Vincent later today is going to go through ashwagandha, which we had 3 lots from 3 different suppliers. But for the rest, and for our purposes of just trying to do assay toolkit validation or suitability rather, we're only doing 1 lot at this time. Okay, we have a few more minutes for questions. So one that came in said, while you are not endorsing assays, won't that occur when you articulate a strategy?
I'm sorry, Connie, I'm not sure I understood the question. I apologize. Can you reread it?
Yeah.
We're not endorsing specific methods, yeah.
So I guess it would come up. I think more what we were trying to avoid is one from one company. I mean, in some cases that might be the effect, but we're not trying to make something that's not open to a lot of groups. In cases we're trying to find assays that are maybe available from a lot of different contract labs or have really established methods that labs could follow. So I hear your point that came in from Stephen, but we're just trying not to say one from a particular company is the only thing we're trying to use. We're trying to keep the toolkit open so it's easy for people to use. Okay, another question that came in is, what entity is performing the ID verification for botanicals being studied? And is it the raw material or a finished product that is reviewed?
That might be one you could cover, Connie.
Yeah, so we have quite a few folks on our chemical analysis group helping us out. We're doing HPTLC verification from 2 separate labs, from Alchemist Labs and the HPTLC Association in Europe. And then we're also having labs look for the key constituents to identify and quantify them to make sure it's there. And we also have plans to make sure there's not contaminants. So we have a couple academic labs to help us with the key constituents and trying to go a little deeper, typically using mass spec. And we'll show today a really deep dive into one on ashwagandha, and it's the raw material. There's a couple.
And I think that's a great. I think that's a great example of the importance of the pharmacognosy group as well, sort of providing us some insight as to what should we be testing. So I think a lot of them are, raw plant material. We specifically identify green tea extract. So I think there's in some cases the preparation does matter, but that's sort of the import of the input from our working groups.
Thanks, Cara. And for the sake of time, we'll go ahead and move on to our first keynote. So I'm going to turn it over to Cynthia Ryder to introduce. Thank you so much, Cara.
Hi, everyone. I'm Cynthia Ryder, a toxicologist at the National Toxicology Program at the National Institute of Environmental Health Sciences and co-chair of the Botanical Safety Consortium. It is my pleasure today to introduce our first keynote speaker, Dr. Ulrike Grienke. Dr. Grienke received her doctorate in pharmacy from the University of Innsbruck in Austria, where she investigated anti-inflammatory natural products using computer-aided methods. Following her postdoctoral training, Dr. Grienke was engaged in work on identifying the cardiotoxic potential of hERG blocking natural products and studying the pharmacokinetic parameters of hERG blocking alkaloids. She is currently a senior scientist at the University of Vienna in Austria, where she is involved in discovery of bioactive constituents from natural sources using NMR-based biochemometric approaches. Thank you so much for being here, Dr. Grienke.
Thank you very much for this introduction. And also thank you for inviting me for this talk online in your online annual meeting this year. I'm going to talk about our hERG screen project, enhancing botanical safety by cardiotoxic risk assessment. When we speak about hERG, what is hERG? I think most of you know, but just in case, I'm going to repeat a bit the basics. hERG stands for Human Ether-Agogo Related Gene Channel. This is a potassium channel, which has a key role in repolarization. Actually, you can see here a normal ECG recorded in black. And especially the QT interval that you can see here is influenced by the hERG channel. The QT interval is a measure for the duration of ventricular depolarization and its subsequent repolarization. So what happens when you inhibit the hERG channel? The hERG channel can be inhibited by drugs or by other substances, and the effects are pro-arrhythmic actions and even can end up in torsade de pointe, which leads to cardiac death in the worst case. So it's not really something healthy that you want to have. This problem is well known, and especially in drug discovery, it's systematically tested. So all drug candidates have to go through hERG testing. And it's routinely evaluated for drugs, also for drugs that are on the market. It's continuously re-evaluated. But what is not evaluated is the hERG blocking potential of botanicals. It's not evaluated. And this made us a bit worried, and this is why we launched a project that we called the hERG Screen Project, hERG-related risk assessment of botanicals. It was an FP7 project. I don't know if you know about FP7. It was an EU-funded project that took place between 2012 and 2016. And we tried to address the urgent need to systematically test natural products and botanicals and evaluate their cardiotoxicity. And the aim was, of course, to improve consumer safety, especially regarding commonly consumed botanicals and dietary supplements. And this is why we established a network between European, South American, North American, South African educational and research entities, all participating in this project. Here you can see an overview of the hERG Screen Consortium. We were 9 project partners from different continents, as I said, and we had almost 60 secondments taking place. So it's an exchange of people, an exchange of scientists going from one partner to the other to do experimental work, to get training, especially for young scientists, This also included teaching or the organization of workshops that we had in Austria, in Greece, in Brazil, and in the end again in Austria. This project was coordinated by Judith Rollinger, that you can see in the middle here. I don't know if you can see my cursor. And the project was managed by Christina Meyer. She is on the left in this picture. And what was our aim, or what was our workflow for the hERG screen project? So we decided to specifically select commonly consumed botanicals that are not regulated so far from the involved continents of our project partners. So in total, we selected about 650 species, plant species, or even mushrooms that were collected in a first step. In parallel, we also performed an in silico part of this project. We created pharmacophore models, which should then be used for virtual screening of natural product databases in order to filter out potential hERG blocking constituents from virtual databases. Starting with this in silico part, I would like to just highlight how the approach was here. Here you can see the 3D alignment of known active compounds. Active is a bit strange name here to say active. Actually, the activity was the hERG blockage, some undesired activity. And we took known hERG blockers, aligned their structure, and then identified common features. And with these common features, we built up a computational model, added spatial restrictions in gray. You can see them here on the right side, where no functional groups should be due to the macromolecule. And then we used these models for virtual screening of natural product databases and received hit lists with potential hERG blocking constituents. So these were all predictions. In order to evaluate our predictions, we performed an experimental validation of these results, and you can see here 20 out of 50 virtual hits had IC50 values between 0.1 and 2.8 micromolar. So they were quite active on the hERG channel. So this model turned out to be highly suitable to identify or to predict hERG blocking activity for compounds that were not investigated before. On the other side, we chose extracts from, we prepared extracts from the selected species of commonly consumed botanicals. In total, we generated more than 1,200 extracts. And as you can imagine, extract preparation is not extract preparation. Therefore, especially having 9 project partners all over the world, we created a standard protocol for extract preparation, which was done the same way in all the project partners. This is also documented in a YouTube video. I'm not going to show it now because it lasts about almost 5 minutes. So if you're interested, maybe you can watch it later on. It gives you a detailed instruction how we prepared our extracts for the hERG testing. And this protocol we adapted from a recently published paper, where first of all, very non-polar components are removed, and also tannins, which might be interfering with bioassays, were removed. And also we then thought of how to label these extracts, because having a huge extract database, you need to have a good labeling system not to get confused with all the samples. And also this, as you can see here as an example, contained the species name, the organ that was used, the solvents that were used, and also the project partners and batch numbers. So it was easily identifiable through our system. Then, having these first steps done, we continued with an in vitro testing for hERG channel blocking. And also here, we needed some criteria in order to decide which one is a hERG blocker and what is not a hERG blocker. And therefore, we decided, based on literature, to test our extracts at 100 microgram per ml. And we set a threshold of a 30% blockage. Everything that was above 30% blockage was already a hERG blocker. And we followed up which constituents might be responsible for that. So it included then a phytochemical investigation and subsequently the isolation of hERG blockers and also following up on virtual hits from our pharmacophore models. In the end, the constituents or the botanicals were tested in vitro, and for the most active ones, we also performed PK and PD studies within our consortium. From this project, I would like to show you 3 examples that are all based on virtual predictions and where we followed up on the hERG toxicity. First example is a plant called Wakanga africana. You can see here it's a tree which has special fruits and nice seeds, and the seeds and the bark are actually the ones that I used. Why did you select this plant material? We had one of the virtual hits. It's a Wakangine. It's an alkaloid. It's an iboga type alkaloid, which was found as an active hit by our computational model. In order to evaluate it experimentally, we performed an in vitro assay, and indeed Wakangine turned out to have a very low IC50 value on the hERG channel. So Wakanga africana, you might say it's not an important plant, nobody has heard about it, but we then searched what Wakanga africana has as a background. In West and Central African folk medicine, this plant material is used against malaria, asthma, diarrhea, and even convulsions in children or hypertension. It's also used for religious purposes as hallucinogenic and aphrodisiac agent, especially the seeds and the bark. And then we searched a bit online, and we found even online shops where they advertise the herb of the gods, Wakanga africana, root bark, and also seeds that you can just buy over the internet. And it turned out that this plant material is, has an increasing popularity in Europe as a legal high, because it's not illegal to buy it. And the people are using the hallucinogenic effects. What is Wakangine used for? It's also used for the semisynthesis of ibogaine. Maybe you have heard of ibogaine. You can see it's only differing in one chemical position here. Ibogaine is known that, or is advertised that it can reverse addiction to recreational drugs. So it's an anti-drug against alcohol or opiates, and it's also advertised to reduce craving. And interestingly, there are no control studies in humans, and there's a lack of safety and efficacy data, so we couldn't find anything. And we even found an article in this magazine here where it says ibogaine may be the miracle cure for addicts if they can survive the trip. So it's already considered quite dangerous, and also it was listed as a potential cardiovascular side effect causing drug. And it's differently handled in different countries. So in other countries, it's completely unregulated and unlicensed. So it's quite a gray area that we see here. What is now the relevance of our Wakangine results when we transfer our in vitro data to in vivo? So we didn't know about the bioavailability and how the whole pharmacokinetics work for Wakangine. This is why we also performed an in vivo study where we checked the bioavailability, and we saw that actually the bioavailability of Wakangine is quite low. It has a high plasma protein binding, and its character is highly lipophilic. So it can deposit in fat tissue, And this is maybe also dangerous because you never know if it's then accumulating there. And we also investigated one of these commercial products that you can buy online, and we prepared an extract as it was indicated on the package, and we then evaluated the content of workangine, and we saw that this is quite high. So we found about almost 10% workangine in one of the extracts. So it's not really harmless, this substance. The next example that I'm going to present is Cara ipecacuana, which is also called ipecac in a common name. You can see here the plant has nice flowers, and especially the roots are used traditionally. Also here we followed up our virtual predictions. 5 alkaloids from this plant material were predicted to be interfering or modulating the hERG channel. You can see here emmetine, cephaelin, cycotrine, O-methylcycotrine, N-methyl. And also these compounds were then tested against hERG blockage. And at 50 micromolar, all these 5 compounds inhibited the hERG tail currents with varying percentages. And we then performed, for the most active, we then performed the concentration-dependent inhibition assay, and we could also see that there is a really nice concentration dependency of these 3 constituents, and the IC50 values are also in the low micromolar range. What is here the relevance for human health? You might all know ipecac syrup is used as an emetic, especially for children. It's used when they have problems, or if it's gastrointestinal problems. And the syrup contains between 1.2 and 1.5 milligram per ml of total ether-soluble alkaloids, and about more than 90% of them are emmetine and cephaelin. So this is just well known, but what happens if this ipecac syrup is abused or misused, for example, by patients having eating disorders? You can even find on the internet blogs where they propose the 11 best ways to make yourself throw up. So if you take such a medication, which is actually a medication, on a long-term level, or in a state where you are looking like this, I mean, the body is already not in a very good condition, you will also see that the plasma level of these alkaloids increases rapidly, and you have a high tissue accumulation, and it can induce cardiotoxicity. So in these cases, it's really dangerous to take this syrup. The third example that I would like to show you is Nelumbo nucifera. It's the lotus plant, which you also know maybe. Also here we followed our virtual predictions, where 4 apophene alkaloids and 1 isoquinoline alkaloid was predicted by our models to be active hERG blocker. And we also phytochemically investigated this plant, and we then isolated constituents, and we then tested them in an in vitro assay. And you can also see here we have different IC50 values in the low micromolar range. And also this then had to be transferred to what is the background behind it. Maybe you have heard of the lotus leaf diet. It was quite popular even among Hollywood actors to lose weight by taking lotus leaves or lotus leaf tea. And luciferin is known to block fat absorption, even to inhibit the pancreatic lipase, and also it inhibits fat accumulation, inhibition of adipocyte differentiation. This is all known for luciferin. But we also found out that it has an in vitro activity on the hERG block, on the hERG channel. Then we asked ourselves, how harmful are lotus products, especially dietary supplements which are not regulated, which have no requirements about maximum concentration of constituents inside. So we just randomly selected 5 products that are used for weight loss, and we quantified hERG blocking alkaloids. So I don't want to confuse you with all the numbers here. Basically, what you can see is that we analyzed 5 products that you can see in different colors here. And we then calculated the amount of alkaloids present in these products in the daily recommended dose. So you can see, let's take nuciferine, this alkaloid, where we have quite a high amount in one product. And for example, in product 4, we have nothing at all detected. So you see there's a complete variation of constituents present in the different products. And this is not surprising because there is no regulation, and basically this just depends on where the samples come from. What about pharmacokinetics? Nuciferine was previously quantified in rat plasma and tissues after oral and IV administration. And also here it was observed that there's a rapid distribution, extensive tissue uptake, poor absorption into the systemic circulation, and also low bioavailability. But it distributed into brain, liver, and adipose tissue. So in general, we should pay attention that lotus products are non-standardized dietary supplements, and depending on the condition of the person who takes these dietary supplements, it might also cause cardiotoxic issues, especially when the person already takes some medication or there are some interaction with other drugs. You never know. So what was the conclusion of our hERG screen project? At the end of the project, we performed a huge literature survey, including our results from the project, which are published in this review here in Natural Product Reports. And we performed a huge data analysis. We took all the hERG channel interacting compounds that were available from the public domain and also from our project, about 200 compounds, and characterized them into alkaloids and non-alkaloids. And then we also performed the classification into strong, moderate, or non-hERG blockers. And what we can see here in this graphic is that hERG blockers seem to be concentrated among the group of alkaloids, especially here in this red part. You see there are strong hERG blockers among alkaloids, whereas the non-alkaloids are, there are less strong hERG blockers among non-alkaloids. And what does this mean? Can we take some information from this? We performed also a principal component analysis where we analyzed the lipophilicity, the size, and the aromaticity of the compounds analyzed for their hERG blockage. And interestingly here, when you look at this graphic, you see the small dots here, also color-coded, strong hERG blockers, moderate hERG blockers, and no hERG blockers. You cannot really identify clusters here. So it's not easy to distinguish between blockers and non-blockers. So it's not easy to just say, okay, just because it's an alkaloid, it must be a hERG blocker. But there is a tendency that more hERG blockers are in the lipophilic region, but that's basically all that we could deduce from this analysis. So what is now the overall outcome of our project? As I said, we produced more than 1,000 extracts of commonly consumed botanicals from 3 different continents and tested them all for their in vitro hERG blocking activity. Interestingly, only 2% showed a reduction of the hERG channel peak tail current by more than 30% at 100 microgram per ml. This indicates that there is really a low risk for hERG channel related toxicities by commonly consumed botanicals. But we should not say everything is okay and there is no risk at all. We should be very careful because our results, of course, are just in vitro results done in an artificial environment and not in vivo results. So it's very preliminary, but it gives a good impression from the in vitro side. And of course, there is no direct translation to increased risk of QT prolongation. So this is all a bit still to be evaluated. And in the end, I would like to thank the whole hERG screen team, especially Judith Hollinger, the coordinator, Christina Meyer, the secretary, project organization, and Jadel Kratz for performing the in silico part of these studies, preparing pharmacophore models. And thank you again for the invitation to participate, and I'm happy to answer any questions.
Great, thank you so much. That was a fantastic talk. Please, if you have questions, we have a few minutes. One question did come in already. I think you addressed it, at least at the beginning, a little, but Peter Hoffman asked, some compounds appear very lipophilic and may stick to the walls. Did you measure bath concentrations to test whether the nominal concentrations were achieved during your in vitro test?
You mean the walls of the Eppendorf tubes, I guess, or?
I believe so, what the exposures were done in, yes.
Yes, so the testing was performed in our, with the project partner doing pharmacology, and there they had a protocol to be followed. I don't know the exact details by heart anymore, but there were also precautions done that there is no sticking to the wall or other interfering problems happening during the testing.
Thank you. I had a question about cell type considerations. I saw on one slide it was the HEC293s. Have you
Yes.
done any of this work in cardiomyocytes?
This is a bit problematic because they're much more difficult to handle, and this is why the HEC cells were chosen, because we needed a stable model for that large amount of samples that needed to be tested, and also this was not very easy. So yeah, we trusted our pharmacology partner who performed the testing, and they preferred the HEC cells in this case.
Great, thank you. I don't see any other questions in the chat, so I'm going to thank you again for your talk today, and thank you for giving us this update on your work. Thanks so much.
Thank you.
All right, so our next speaker today is going to be Dr. Vincent Sica. Vincent Sica is a senior scientist at Procter & Gamble, who along with Dr. Tim Baker has been heading the chemical analysis efforts on our pilot botanical ashwagandha root. So I'm going to give Vincent, oh, you already have the ability to share, or is that still before?
There we go.
Great, and you're the presenter now, great.
Do you see my screen?
Yes.
Okay, perfect. Thank you.
But you're not in the presentation mode yet.
Is it now coming up?
It's still not.
Do you see my?
Like we can see the icons. There you go, perfect.
Oh, I got you. Sorry about that. Well, great. Once again, my name is Vincent Sica, and I'm a senior scientist at Procter & Gamble. And today I'm going to be discussing the chemical analysis of ashwagandha. So at Procter & Gamble, we characterize several botanicals. Botanicals are used in many of our products, and over the years, we've encountered several challenges when trying to look at the chemistry of these complex mixtures. Fortunately, we've been able to assemble an analytical platform that overcomes many of these challenges. We typically apply this platform when working closely with our toxicologists in our safety department to help guide their safety assessment of the use of a certain botanical. This typically starts with them providing us with a threshold of toxicological concern, or TTC, which helps guide our chemical constituent identification process, or CCID. For the pilot for the Botanical Safety Consortium, Ashwagandha was selected to showcase our analytical approach. And it was largely selected due to the reliability of materials from well-known suppliers and the availability of a USP standard for authentication. In fact, we've looked at 3 different suppliers and compared them to the USP standard, and they were all essentially equivalent. That being said, I'm hoping today that I can convince you that if you were to take our approach towards less known suppliers, you would still be able to perform a chemical analysis and determine authentication and adulteration studies, look at supplier and extract comparisons, and guide toxicological assessments for the clearance of the safety of a botanical. Unfortunately, there are several analytical challenges when trying to look at botanicals. Many of them were discussed earlier today. Botanicals are, of course, complex mixtures with a lot of different compound classes. There are a wide range of different constituents, and even ones that are very closely related that are difficult to distinguish. What you're seeing here on the slide here is a chromatogram of ashwagandha, and chromatography is essentially our ability to take those complex mixtures and try and separate them out so we can ideally look at them one at a time to identify and quantify them. Unfortunately, that's not always that easy. Once again, as you imagine, we encounter isomers or those closely related compounds. We get co-eluting compounds. So based on the chemistry, our separation is not always able to separate them out. And we have compounds that go through the chromatography at the same time. So just because in this chromatogram you see a lot of peaks, just because you see a single peak, it doesn't necessarily mean there's only one compound there. Additionally, there's a large dynamic range. Inherent to these botanical extracts, there are some compounds that are in higher abundance than others. Just like you see on this chromatogram, some peaks are really tall, some are really small, and that becomes very difficult when you're trying to quantify these complex mixtures. And of course, there's literature concerns. Sometimes literature could be limited or inaccurate, especially when looking at lesser known botanicals, trying to find out their toxicological effects or even what compounds are reported to come from there. It can be difficult when it's just not there available in the literature. Similarly, there can be a lack of available standards. Standards or authenticated chemical materials which help quantify and identify the constituents can be very limited. Even for very well characterized and very well characterized and well-known botanicals, there may only be a handful of available standards, and this can certainly complicate a chemical analysis. And then, of course, there's the inherent variability from the botanicals themselves. A lot of variability. How is it extracted and processed? What time of year was it harvested? What's the stability of those constituents? All of these other factors all play a role in making this such an analytical challenge when looking at botanicals. Fortunately, we've assembled an analytical platform that overcomes these challenges. Once again, this all starts with our chromatography, or our ability to separate out this complex mixture, ideally enough to where we can look at them by their detectors one at a time. We use ultra-high performance liquid chromatography, or UHPLC, to separate these out, and then send off these individual analytes to 3 different detectors. The first detector is our photodiode array detector, or PDA or UV detector, and this provides a lot of supportive information. We get a little bit of qualitative and a little bit of quantitative information, and actually, most importantly, it's a non-destructive technique, which means it's kind of free information. It can pass through the detector, give us information, and then we can pass it along to additional detectors. Our next detector is the charged aerosol detector, or CAD, and this is what we consider our quantitative detector. The CAD is a semi-universal detector, meaning that if you have more of a constituent in a mixture, you'll get a larger peak. If you have less, you'll get a smaller peak. And this is all independent of the actual chemical structure. And this is a really powerful tool that honestly makes this all possible, because we can actually take a botanical mixture without knowing at all what is in that mixture, and quantify all the constituents using totally unrelated standards. This is how we overcome some of that limited standard availability or authenticated chemical materials, because we can use this approach to quantify almost everything in that mixture. And then finally, our third detector is our high resolution mass spectrometer. And this is where we get an abundance of qualitative information. We can not only look at the analytes in a variety of ways, positive and negative ionization mode, but we can also perform fragmentation studies where we essentially take that compound, break it into smaller pieces, and then like a puzzle, build it back up to determine its structure. Now, while our mass spec is typically our most sensitive detector, it's also the most highly dependent on the chemical structure itself. Small fluctuations in a structure can actually greatly affect how well it's detected and how big the signal is on the chromatogram, which is why we really rely heavily on that CAD for our quantitative ability. So here you can see a snapshot of a single injection of ashwagandha onto this platform. And while you can see overall they generally look fairly similar, there are some differences. And what's really important here is that these 4 different looks of each sample allows us to ensure that we are not missing any of the chemistries when we perform our analysis. So that when we go through a botanical and characterize the chemical constituents, we're able to provide our toxicologists with all the information because of these several different looks, all this mutually supportive data. Once again, though, we really do key in on the CAD for our quantification. If you look at that peak between 30 and 40 minutes, the largest peak there, you'll notice that it's not always the largest peak on some of the other detectors. If you consider our high resolution mass spec in positive ionization mode, which often gives us the most qualitative information, it's barely in the top 5, whereas the CAD determined that it is actually our most abundant constituent. Conversely, if you look at the peak between 50 and 60 minutes there, it's responding very well on our mass spec, but on the CAD side, it's saying that it's actually a very low constituent. So how do we use this platform to help support toxicological assessments? Well, once again, we work closely with our safety department, and they typically provide us with a threshold of toxicological concern, or TTC. And what that essentially is they draw a line and they say anything at a lower concentration than at or below this concentration is such a low amount that it's not considered a toxicological concern. Anything that is above this line would then need to be quantified and identified so that they can properly make their assessment. Now, it's important to note that just because it is above this threshold does not mean it's a concern. It's just something that safety would like to investigate deeper to understand what that chemistry is. In the case of ashwagandha, I went through and I integrated all the constituents. Oh, I'm sorry. Unfortunately, that line that safety draws is typically a very conservative approach, and it's way down here. So I end up resulting in looking at almost everything that I could detect. When I did go through and I integrate all the constituents in this sample, I determined that there were 46 CAD peaks that were above this TTC threshold. Collectively, those 46 peaks made up a mass balance of 99%, meaning they count for 99% of the materials that I prepared. That remaining 1% is a compilation of the constituents that were below the TTC, that were no need for concern. So the next part is I take those 46 CAD peaks and I perform my chemical constituent identification, or CCID. This all starts with determining the number of constituents that are underneath each of those CAD peaks. So once again, just because there are 46 CAD peaks does not mean there are only 46 constituents due to those co-eluding peaks. So I go through, I'll look at the mass spectral data that corresponds to each peak and determine the number of constituents. I then use those accurate mass measurements to determine the molecular formula. And what's really great about this is that very early on in this process, I'm able to take the molecular formula and already begin my literature searching and determine what compounds do I expect to see in this botanical. I then consider the orthogonal data, the UV data, the fragmentation, the retention times, and I then begin to use that information and to mine literature and databases to determine potential structures. Once I have a list of potential structures, I could then categorize them by a level of confidence and how confident I am in that identification. So let's go through an example here. If you remember on one of the earlier slides, I showed you that largest CAD peak. This is the corresponding mass spectrum to that CAD peak. And as you can see here, there are several signals, which you might think indicates that there's a lot of co-eluding peaks. It's my job to go through each one of these signals and determine whether they're related to each other or not to describe a single entity. And as you can see here, many of these other signals actually all correspond to a mass or a constituent with a mass of 783. So some of them are in-source fragments, some of them are adducts or artifacts of the instrument, but they all correspond to a single constituent. I can also go through and determine which ones are not related to these constituents, such as this one here at 489. So I do in fact have a co-eluding constituent, but it's not as many as it may look at a first glance. Because we're using accurate mass and high resolution mass spectrometry, and it's accurate out to the 3rd and 4th decimal place, I'm actually able to use this number and determine its molecular formula. So in other words, the number of carbons, hydrogens, oxygens, sulfurs, nitrogens, what have you. I can then take that molecular formula and begin searching the literature to come up with potential structures, ideally ones that are known from coming from ashwagandha. I could then perform fragmentation studies, and you can see 2 examples here, where we break that constituent into smaller pieces, and then like a puzzle, determine their molecular formulae, determine potential structures, and piece it all back together to see if it matches the structure proposed in literature. Once I have a structure that I feel confident in, I bucket it in a level of confidence. And there are essentially 4 criteria I use to bucket these. The first one is, does the molecular formula make sense for the high resolution mass spectrometry data? Does the fragmentation support literature or what I see in a database or even manual interpretation? Does the literature support this presence of this compound in the plant of interest? So does this compound, is it known to be coming from ashwagandha? And then finally, if I'm able to get a hands on a standard, does the retention times match with the constituent I'm seeing in my sample? So my highest level of confidence is the case where I can actually buy a standard. I can run it on the same platform and say, yes, the retention times match, my accurate mass matches, the fragmentation matches, and it's known in literature to come from ashwagandha. My next highest level of confidence we consider matched. And that's usually a case where I can't purchase a reference standard. But the accurate mass supports the formula, the fragmentation matches a database or literature, and the literature supports that this compound makes sense to be coming from ashwagandha. My next level is tentative, and it's actually a higher confidence than it may sound, because I have an accurate mass, which gives me the formula. I have fragmentation, which makes sense and is supported by literature or a database or my interpretation. It just hasn't been reported to come from ashwagandha yet. A lot of the low-level constituents, when we go through, are often not necessarily reported, and a lot of those end up being a tentative match. So I'm still very confident in the structure, or at least the substructures, maybe not the connectivity, but it's still a high degree of confidence to supply to a toxicologist. And then finally, our lowest level, we consider partial. And at the very least, I have a molecular formula, but typically I have a lot of fragmentation information that can help determine some of the parts of that structure. For instance, in this example here, I know there are 4 sugars that attach to that species. I just may not know what that core structure is or exactly how it's coordinated, but it's still a lot of information for a toxicologist. For instance, sugars are known to have a detoxifying effect, and a toxicologist can make their assessment even with some of this limited information. So what happens from here is I then go through and I take those 46 CAD peaks and I tabulate them with the number of constituents under each peak. I list their retention times, their accurate mass measurements, their proposed identifications with molecular formula and my level of confidence, the proposed structure, the quantified amount that I get from the CAD for the amount per day per dose, and then some comments to determine how I came up with some of these, my level of confidence. A toxicologist can then go through these data, and based on the chemicals, they can look at the hazard data and proposed exposure and begin making a safety assessment. And then these data can inform that safety assessment and help guide the need if there are any additional classical safety studies that need to be performed. So in conclusion, hopefully I was able to convince you that our analytical provides an abundance of mutually supportive data. and that we can use these data to perform a comprehensive chemical assessment of a botanical that can help support safety, as this data can help support a toxicological assessment and guide any future steps to ensure the safe use of these botanicals. Thank you.
Thanks so much, Vincent. If people have questions, please go ahead and put it into the question box. One question came in from Nick Oberlees, and he said, very nice presentation. I understand you only needed to look at the 99% of peaks. However, do you ever scan anything below the TTC just to see if there's anything obvious, or do you simply rule them out?
We do. So while the TTC is already a somewhat conservative approach, we typically build in an uncertainty factor on top of that, so maybe as low as 25, 50% below that, to ensure that, if there is some variability, and as I explained, how it was processed or harvested and whatnot, that we're ensuring that we're covering everything that could be safe. So typically when I supply this to safety, I am going below that threshold just to ensure that we're looking at everything. Sometimes if there is a specific compound of interest that they know would fall below the TTC but still pose a threat, I can target those and look for those as well. So we do sometimes go below there, yes.
Great, and then question, there's actually two from Saramia. I'll read the first one. How did you confirm the concentration of a given peak or analyte in those cases where there were co-eluting peaks in the CAD?
So that's a great question too. So it's difficult, right? We really can't. So typically what they'll do is they'll make a conservative measure and assume that each of those co-eluting peaks are the maximum amount of my quantified amount. So if I take a peak and I quantify it to say 90 micrograms per dose, if there's 3 peaks, our toxicologists would assume that each one of those are at 90. So it's another level of conservatism in there. If we really needed to know it, and we would ideally try and get a standard if available, or maybe try different chromatograph approaches and target those individual constituents. But typically they'll just assume that it's worst case scenario.
Thanks. So Rami, I don't know if I understand your second question. I'm going to go to Nick's real quick. So how many ended up in the tentative category?
That's a great question. It's actually still ongoing, so I don't have the final count for it yet. I apologize. I don't have the answer for this one yet.
That's okay. And what parameters were used to define the TTC? Okay,
sure. So toxicologists would be able to explain this much better than I could. But essentially, there are different, like I said, they're established in literature, Kramer class, and I think Kramer class 3 is the one that we use for this study here. And it determines, I think it says 90 micrograms per day is the threshold. Of course, it depends on dosing and whatnot. But yeah, I'd have to look in the literature, but Kramer class 3, I think, is what was used for this. And that usually pertains to developmental and reproductive toxicology, I believe.
Great, there's a few more. We're a little short on time, so I'll try and get through these. Have you seen some variabilities in constituents depending on the subspecies, countries, where it's coming from, or the extraction approach?
Yeah, so extraction approach, absolutely. We've looked at a variety of botanicals where we looked at different extraction methods, and that certainly plays a role into the levels of constituents. Regions, we haven't done I'm trying to think of an example where we've really looked at them specifically targeting different regions. We've seen slight fluctuations in a couple of examples that I can think of, but we haven't done a full study to show how much that differences are. So it's usually a little bit of a relative difference, but qualitative, they're usually pretty similar.
Thanks, Vincent. So we're at time, so we're going to go ahead and move on. Thank you so much for your chat today. There are a few more questions and we can follow up with that. So our next speaker is Dr. Michael Lawless. He is a senior principal scientist at Simulations Plus. Michael, along with Dr. Annie Luman and Dr. Esther Hagebrooks and the systemic toxicity team, have been working to pilot modeling for botanical constituents of interest for the consortium. Thank you so much, Michael.
Thank you, Connie. Hello, everyone, and thank you for attending the 2021 Botanical Safety Consortium annual meeting. I want to first thank Connie Mitchell and Michelle Embry for the opportunity to present our work on modeling botanical constituents, and all the wonderful work that they've provided for this project. So myself and my co-authors are part of the Systemic Toxicity Working Group, and we're trying to model absorption, distribution, and metabolism of botanical constituents. This next slide is simply an outline of the talk. So first I'm going to just talk about what's happening in vivo when you take a botanical orally, talk about special considerations for botanicals, in silico and in vitro to in vivo extrapolation, and then predicted ADME properties of botanical constituents. In the next section, I will talk about pharmacokinetic simulations of all the botanical constituents. So this is a little bit of a less rigorous approach than the full PBPK models that I'll talk about in the very second to last section, and then talk about future studies. So this slide tries to explain what's going on in vivo and relate it to the properties we get out of the modeling. So a botanical is going to be, obviously ingested and go into the stomach where it will start dissolving. The stomach pH is anywhere from 2 to 4. So the pKa, the compound, is important. Also, depending on if you take that pill in a fed state or a fasted state, the compound will stay in the stomach a different amount of time, and we can take care of that in the simulations. Since the pH in the stomach is very low, we have to consider pKa, the compound, and its dissolution process, so the solubility versus pH. And then also as the compound works its way down the GI tract, the pH changes, so it gets closer to 7 in the other compartments in the GI tract. We calculate biorelevant solubility, or we can measure that in vitro. So this is solubility in fasted and fed simulated gastric and intestinal fluids. And then precipitation kinetics. So is the compound dissolving and then reprecipitating along the GI tract. Only the portion of the compound that's in solution can be absorbed into the gut wall. So anything that's not in solution in about the 24 hours it works its way down the GI tract would be excreted in the feces. Once it reaches the enterocytes in the gut wall, we term that portion the fraction absorbed of the compound. There are also enzymes and such as cytochrome P450 and UGTs in the gut that can metabolize the compound, so clear in the gut wall itself. In order to get into the gut wall, you have to consider transcellular and paracellular permeabilities. So transcellular permeability is the compound coming in one side of the cell and going out the other side of the cell. Paracellular permeability is relevant for small molecules that can kind of go between the cells and be absorbed. Log D is highly related to the absorption, so we need to have a good log D versus pH profile of the compound. Gut extraction is related to the metabolism. And then there are transporters in the gut wall, which will either, which could influx the compound, so transport it from the lumen to the gut wall. And then there's also transporters such as PGP that will try and prevent compounds from getting or being absorbed. Next, the compound would traverse into the portal vein. We can also calculate the fraction of the dose in the portal vein, which is different than the, which can be less than the fraction absorbed if we're considering metabolism in the gut wall. Next, the compound will be delivered into the liver, where an additional amount of metabolism can take place. And this is also a main metabolizing property once you get into systemic circulation. So we have models that will predict liver metabolism from in vitro experiments, or of course we can always measure that in vitro. Hepatic uptake is transporters that would transport compounds from the blood into the liver cells. And then biliary excretion is a way to clear the compounds. So once it gets on the other side of the liver, it goes into systemic circulation. And when it reaches systemic circulation, this is called the oral bioavailability of the compound. I also term this percent FB. It's not to be confused with percent FA. So typically oral bioavailability can be measured in an in vivo experiment where you've dosed the compound IV, and then you've also dosed it orally, and you take the ratio of the area under the curves. So typically out of an experiment, you can pretty easily get the fraction in oral bioavailability, but you have to perform those two experiments. This is a little bit more difficult to measure in vivo. So once it gets into systemic circulation, it can be distributed into the tissues. So we need to consider that. That's the real power of the physiologically based pharmacokinetic model, is that we have tissues defined in the model. Plasma protein binding and blood to plasma ratio kind of represent how much of the compound is free to traverse some of these into these different tissues. And then we're considering systemic clearance here, which would be both from the liver, enzymes in the blood, and clearance through the kidneys. So what we have in the end, what we're able to model in the end is the dissolution of the compound showed in this red line here in this example, and it corresponds to this axis over here on the right. So here we're saying that the compound dissolves very quickly, and then as it starts to work its way down the GI tract, it precipitates a little bit and then slowly dissolves again, going into past this phase here. The blue line, or the cyan line, represents the fraction absorbed of the compound, and then the green line is the systemic circulation. And the difference here is metabolism in the liver or metabolism in the gut wall. I didn't consider that in this particular model. And then they all kind of work together to give you the CP time curve, which is the plasma concentration here in milligrams per milliliter across the time slot. So we can easily get things like the fraction absorbed, oral bioavailability, Cmax, which would be the maximum peak here, Tmax, the time of the max, and then the area under the curve here. And I just show area under the curve to infinity. You can see that this compound's not totally cleared after 24 hours. So the area under the, going from 0 to infinity is going to be higher than just the area under the curve for this 24-hour period. People have discussed this quite a bit today, but special considerations for botanicals. They're mixtures of many compounds. We don't know the exact composition always. And components can affect absorption and metabolism of other components. So for example, if you have a compound that's a CYP inhibitor and you co-dose that with a compound that would be cleared by a CYP or metabolized by that same CYP, then that pathway is blocked, so you would get a higher exposure of that other compound. And that can occur within the botanicals themselves or with another drug that you're potentially taking. So you can also consider transporter inhibition and inductions of both of these proteins here. In vitro physical, chemical, and metabolic components may not be known for the compounds. And then in vivo data is potentially not largely available. And I've learned a lot just from the talks today about different journal articles, maybe where we can extract more of that information. We're going to be creating models. The PBPK is a model in itself. So we have to remember what George Bach said in 1979 publication, that all models are wrong, some models are useful. So we know that we can't represent nature perfectly with equations. We have to make approximations and assumptions, and our goal is to have models that are reasonable. We're never going to get a perfect model. But reasonable doesn't mean poor. It means they're good enough to satisfy requirements. And so what we're trying to do is use these models to learn more about the compounds and then progress them into different experiments potentially. So mainly what I'm going to talk about today is using in silico predicted properties to create the inputs for the PBPK model. So we're going to take a 2D structure of a molecule, and we're going to predict various parameters here and use those as parameters to input to a PBPK model. We're going to use a program called ADMET Predictor that uses machine learning models to predict these properties from the 2D structure of the molecule. And then we're going to plug that into a physiologically based pharmacokinetic model. And you know, the inputs here can come either from in silico predictions, which I'll talk about today, or in vitro measurements. And that's a very important aspect of the model, progression really. So it will get the time curves that we talked about on the previous slide. And the goal is to reliably and efficiently utilize PBPK modeling to assess safety of these botanicals. We're going to use the Advanced Compartmental and Absorption and Transit Model, or ACAT model. So it's a 9-compartment model starting in the stomach and it's working its way down into the ascending colon. Botanicals mostly are in an undissolved state, and they can go between that and the dissolved state and then transfer to each one of these compartments. So it's very complicated mathematical equations that need to be solved because as a, you know, one portion of the compounds coming into a compartment, it may be going into the dissolved state and then also traversing to the next form, and then it can be absorbed into systemic circulation, where we'll use a PBPK model to analyze that. So a PBPK model is defined as different compartments within the model. So here we have adipose tissue, heart and kidney organs, and then a blood flow kind of connecting all these and circulating around through the various organs. Each tissue or organ is defined as a vascular space. So this is the blood flow across the tissue, an extracellular space and an intercellular space. And for each component or tissue has a specific volume or specific size, a blood perfusion rate or blood flow rate, certain expression levels of enzymes and transporters, and then the fraction of lipids and proteins. And this is important for looking at the free versus the bound species of the molecule. And then these are kind of combined to create the tissue to plasma partition coefficient, or KP. So KP, the higher the KP, the more the compound is sequestered in the tissue versus the plasma. For example, it was mentioned earlier that lipophilic compounds would be absorbed into adipose tissue and can stay there. So they would have a large KP, or KP above 1 for adipose tissue. And it's all in equilibrium together. So just a few of the inputs, as I mentioned, pKa, solubility, biorelevant solubility, log D, human effective permeability. So this is a model of how the rate, it's measured in centimeters per second, how fast the compound would get into or be absorbed or permeate the jejunum tissue. We mentioned both these. And then for intrinsic clearance, we can either use human or rat liver microsomal clearance or hepatocyte clearance in the models to represent that. And the overall clearance is combined. The in vivo clearance also takes into consideration the plasma protein binding and the ratio of blood to plasma concentration ratio, along with the blood flow through the liver. Our models consist of 33 artificial neural networks, so it's not just a single model, it's a kind of an ensemble of 33 artificial neural network models, each with a slightly different prediction for the molecule. And then these are, you take the average to give the output of the actual model. We also create something called regression uncertainty, and it's based on the variance of the 33 models. And this affords us a lower and upper bound to each one of the predictive properties and they're within 1 standard deviation. So in this example, we have the log P, which would be the mean value, then an upper bound and a lower bound that's based by analyzing the data for this. And then in certain PBPK models, I'll might use the average values for all of these and then create different records that would have the lower bound, for example, for the log P and the average of the others, and take a look at how that causes a variation in the different parameters. This is a list of our 13 botanical ingredients. And I want to thank Connie and Michelle for putting together this initial list. Now, these are just the number of compounds where we could create a structure for this. So we need a structure, obviously, to model it. And there are typically more constituents, but the exact chemical hasn't been identified yet. So overall, we're looking at 250 compounds from these 13 botanicals. The largest class is comfrey, and then this class here is the smallest with just 3 compounds in there, but they're all acids. And then some constituents are found in more than one botanical. So if you just look at a unique list, there's 211 unique compounds out of the 250. So the next section that discusses ADME properties. So here I've plotted ionization in different forms here, and then the number of acids and bases in the different botanicals. So if we look at this plot first, most of the compounds, about 60%, are neutral. So they don't have any, they're not ionized at pH 7.4. Another 40% is split equally or approximately equally between acids and bases, 20% each, and then there's one compound that's a Zwitter ion. As far as the number of acids in each botanical, Usnea has the most acids. Acids can sometimes cause liver toxicity. I know that's a kind of a broad statement, but I certainly know of some acids that are involved in liver toxicity. As far as the bases, blue coash has the largest number of bases, followed by ephedra. And these compositions kind of can affect dissolution. So in the stomach, where you have low pH, a base is going to be highly ionized, where an acid will be less ionized. So dissolution for acids is lower than bases typically. A common thing to do for medicinal chemist, if he has a low soluble compound, is to add a basic group into it to try and improve the solubility of the compound. The acids, while they might not dissolve in the stomach, then when the pH gets closer to 7 in the GI tract, then they're typically ionized if the pK is below that, and then that increases their solubility. This next slide looks at lipophilicity, aqueous water solubility, and human jejunal permeability. And remember, all these are predictions again. So for lipophilicity, typically for drugs, you would want to keep the log P below 5. Here you see compounds that are above that and going up already to a lipophilicity of 12 for this. These compounds are typically going to have low solubility, so they'd be associated maybe with this part of the dust distribution for water solubility, but they typically have high permeability. So maybe these are the compounds up there at the higher end of the permeability scale. On the other hand, these are hydrophilic compounds or water-loving compounds. Typically, they're going to have high solubility, but typically low permeability. As far as permeability is concerned, you're going to get low absorption if the PEF value experimentally is 0.3 times 10 to the minus 4 centimeters per second. So you really have to only concern yourself with low PEF values. Once it gets above 0.3, then typically your fraction absorbed is above 50% for those, depending on, again, on the solubility and other factors. Once we reach a PEF of 5, typically that's not going to increase the fraction absorbed any. These are going to have high fraction absorbed, but they're going to be probably low solubility compounds, so that may be affect them too. All right, so in this next section, I'm going to talk about high throughput pharmacokinetic simulations. So these use a bit more approximations than the full PBPK model. So we're going to do a dosage form that's an immediate release tablet. We're going to use a 70 kilogram subject. We do use the full ACAP model for the absorption. We don't consider gut metabolism or active transport of the compounds. And then we're only modeling linear clearance. So the enzymes aren't saturatable, which can be an issue if you know, it has a low Km for its particular enzyme and it's dosed in a higher concentration than that. Okay, so in this slide, I'm just looking at the fraction absorbed versus the oral bioavailability. I'm doing a 100 milligram oral dose, and I'm using the predicted hepatocyte clearance. We can use liver, or we can use microsomal or hepatocyte clearance. When I perform both those kind of computational experiments, I got lower fraction absorbed for the hepatocyte clearance, and that's why I'm showing it in this slide. And this is something we learned from the trying to model the in vivo PK data, is that typically we kind of overestimate the exposure of the compounds. So using something that predicts higher or faster clearance, it seems more appropriate here. So I did it on the 211 unique compounds here. And what you see is the compounds down here in this lower left-hand corner are going to have lower fraction absorbed, below 30%. So typically these are going to be less toxic compounds because they're just being, they're not getting into the system much at all. Up in the upper left-hand corner, these are compounds that have a high fraction absorbed, but they're metabolized. They have high first pass metabolism. So maybe something here you want to consider is, are the metabolites potentially toxic for these compounds? Most of the compounds have low, a reasonable fraction absorbed from the previous plot. But Asian ginseng has a low fraction absorbed typically. So that's illustrated in this distribution plot here, where the red bars represent Asian ginseng, and then the blue bars represent all the other botanical constituents. The red bars correspond to this axis over the right, the blue bars on the left. So you see that many of the constituents in Asian ginseng have low fraction absorbed, below 30% on this plot. And the reasons for that are different for different ones. So I found two examples. Both have predicted fraction absorbed below 5%, so low fraction absorbed. One has very low solubility and the other has very low permeability. So I took these two compounds and I performed a parameter sensitivity analysis. So this value in the middle of both these plots is the value from the prediction of the model, and then we're increasing the value here and decreasing it this way, and then looking at how that affects the fraction absorbed of this compound. So this compound goes from, or this axis goes from 0 to 100% fraction absorbed. And you see that if you were increasing the fraction absorbed, or excuse me, the solubility, or if our model is underestimating the solubility, then that's going to increase the fraction absorbed pretty high for that. If we look at the structure of this molecule, it's got a steroidal scaffold here, and then some kind of hydrophobic side chains. So you'd expect this compound probably to have low water solubility. And then these, you don't have a lot of hydroxyl groups, but those could potentially form hydrogen bonds within the crystal structure and made it harder to break those crystal cells in there and possibly contribute to the lower aqueous water solubility here. This compound over on the right is quite different in its profile. Here, note that this axis only goes from 0 to 12. So even though these curves rise sharply, they would only get up to 12% fraction absorbed, even at the highest permeability and solubility for this compound. And if we look at the 2D structure of the compound, you see a lot of these are sugar rings or hydroxyl groups, so they can make hydrogen bonding networks with water, and so has the higher predicted oral bioavailability, or excuse me, aqueous water solubility. On the other hand, these hydrogen bonds can interact with the membrane, and so they have low permeability because they're interacting. A common thing a medicinal chemist would do if he had low permeability would be to try and decrease the number of hydrogen bond donors and acceptors on the molecule. So now, changing gears a bit, we're going to talk about an ashwagandha study in mice. So this study was from Patel et al., and it was published in 2013. And they looked at with Ferrofin A and with Analide A in a botanical. So they purchased the botanical from Natural Remedies, extracted the roots with distilled water, and came up with this percent composition of these two components. Then their subjects were female mice weighing between 20 and 24 grams. The mice were intergastric administered or oral gavage of an aqueous solution here. And then this is the dose, so about a hundredth of a milligram dose in this fairly large milligram per kilogram of the ashwagandha itself. So this next slide shows the results from the PK study. So in orange we have WLD and in blue WA. And what's interesting about these is instead of just being the concentration going up and then decaying rather quickly, you have kind of the shoulder on each one of the profiles for these two compounds. And then we have the various parameters from a PK analysis here. So what we want to try and do is use PBPK modeling to simulate these results. And there was a lot going into this. Again, I use this method where I take the average and then the upper and lower bounds for different parameters, and then see how it affects the CP time curve. And then in the end, I took the best result from each one of the different parameters, combined that into an overall molecule, and then in the end, I still had to optimize the solubility, precipitation time, and liver clearance. So the first and last one are input parameters that I predicted. This one, the default is 900 seconds in Gastro Plus, but we had to, that's really just a default value, and it would be different for various compounds and compositions here. So in the end, I simulated this as an oral solution as opposed to an IR tablet. That certainly fit the curve or experimental value much better. And then I ended up having the optimized solubility was even lower than the lower bound. And then I used 384 was the optimized precipitation time for this. And so you can see that the blue line here, the CP time curve, fits the points quite well. The correlation coefficient is 0.88. I'd like to see this kind of dip down a little bit and get within this experimental error here, but overall, a very nice fit. And what we're seeing from the prediction is that the compound, while it's totally in solution initially, as it works its way down the GI tract, it crystallizes and then or precipitates, and then slowly dissolves again to give this curve here, or give this shoulder to the curve. So the absorption is kind of prolonged, going out here to about 2 hours. And this is only a 4-hour study in these mice. So from this model, we predict the oral bioavailability to be 33%. And then for the rest of these parameters, we've quite well reproduce these in the simulations. In this next example, I'm going to look at hydrasticine in golden seal. This is from an article from Gupta et al. Published in 2015. So they took 11 human subjects with an average age of 31 and an average weight of 84 kilograms. Each one was given a 2.7 gram dose of golden seal containing 78 milligrams of hydrasticine, and then they took it with 8 ounces of water. The journal article states that it undergoes rapid and extensive phase 1 and phase 2 metabolism, and then they just list O demethylation and demethylation. And I'll show those, what those would be on the next slide. And then these, once they're metabolized, they'd be either glucuronidated or conjugated with sulfates. They also mention CYP3A4, and I think one of the speakers earlier also mentioned potential CYP inhibition studies of golden seal. So on this next slide, this is the structure of the molecule here. So these would be the metabolites that could metabolize these methoxy groups here, along with this N-methylamine. And then this also can be hydroxylated and would be converted to the diol of this. And then each one of those could be have a potential for phase 2 metabolism in creating conjugates for those molecules. So if we look at the CP time curve here, now we're looking at a logarithmic scale over here, whereas the previous one was linear. At 24 hours, the concentration is 1.7 milligrams or nanograms per milliliter. So most of the compound is cleared in one day. I forgot to mention here that in the journal article, they list almost 40 metabolites of this compound that they observed during this in vivo study. And they went out and measured the plasma concentration all the way to 48 hours. So one of the things they mentioned in there was inhibition of CYP3A4 and 2D6. Then they cite another study and specify that CYP3A4 inhibition occurs at 25 micromolar. So we want to try and reproduce this, validate our model, and then take a look at the concentration of hydrasticine and see if it's high enough to inhibit CYP3A4. So in this case, I simply just started with the mean value of all the parameters and then used the upper bound of the human hepatocyte clearance, and that gave this very nice fit to the in vivo data. Now we're kind of overestimating the clearance along this in the terminal phase. So you kind of keep that in mind a little bit, but we really nailed the rest of the points up here, and it fits very well. And these are very low concentrations anyway. So finally, our model gives 100% fraction absorbed, 36% oral bioavailability, so fairly high. First pass metabolism, and then for each of the other parameters, Cmax, Tmax, area under the curve, and half-life, we get within twofold, which is kind of the pharmaceutical industry standard, except for the T1/2, and we're off a little bit on that, and that's always hard to study because maybe if they would have taken another aliquot here, maybe it would have come up a little bit. So we're not too worried about the T1/2 being a little bit off. So I took that model and then I said, okay, what if you take this dose once a day for 7 days in this particular example here? And it reaches steady state pretty quickly. So steady state can be defined in several different ways, but basically kind of reaching an asymptote either along the maximum or the minimum, the trough or the peak, and then others measure it as the area under the curve. So one of our other programs looks at, I think, the area under the curve for a single dose going out to 0 to infinity. If that equals the area under the curve for a specific time, then that is set to reach steady state. Anyway, hydrasting is predicted to be reach steady state quickly, and that correlates with its almost complete clearance within 24 hours. So then now let's look at the concentration of hydrasting in the liver, where there's a lot of CYP3A4. And what I'm plotting here is the concentration in the liver or the compound in nanograms per milliliter. And then this red line represents a 25 micromolar concentration. And you can see at this dose and with this model, these approximations, the total concentration of the liver only gets to about 10 to the third nanograms per milliliter. So it's certainly under the 25 micromolar. And then the unbound concentration showed here in the dots is even lower. So just based on this, it doesn't quite reach enough, high enough concentration. Now, if you took this twice a day, maybe you're talking about, potentially reaching that limit, or 3 times a day. You have to remember that toxicity is related to the dose. Okay, so then let's also look in the enterocytes in the gut wall. So cytochrome P453A4 is also in there. So we use the same model, and we're looking at the concentration in the duodenum, jejunum 1, and jejunum 2. And this is where most of the absorption of the compound takes place. So this is where you'd have the highest amounts or concentrations potentially. And here we're using milligrams per milliliter instead of nanograms. So it looks like we've lowered this substantially. But you can see just on the Cmax, it does touch this even in the jejunum 2. So, is this enough to really inhibit the compound? Yeah, certainly for this time frame, which it is, it appears that it would potentially inhibit it in the GI tract. And then moving on to future experiments. So we want to look at modeling other in vivo studies. I've taken some notes today of things that are missing from this slide, but I'd like to look at milk thistle. This is an article in Drug Metabolism and Disposition in 2008, and they're looking at flavolignans and saying that they're quickly metabolized to conjugates. Annie Luman's already started this study of looking at ashwagandha constituents in rats. Here they did look at PO and IV study, so they were able to measure the oral bioavailability of the compound. And our preliminary studies are reproducing the in vivo profile. Kava, this was an interesting article from Matthews et al. So they took one of the components of kava and dosed it without the rest of the extract, and then they redosed it with the rest of the extract in rats. And the concentration or the area under the curve of Kawane greatly increased when it was administered with the kava extract. So this indicates a botanical interaction to increase the exposure of Kawane. Yohimbine, there's several studies, PK studies in human that we'd like to look at, and then berberine. So one of our kind of a new members to our group, Yitong Liu, actually did some experimental work measuring the PK and in vitro parameters for berberine, published those in 2010, and shows she has started to create a model of berberine in rat to simulate this data. And then we'd like to incorporate more in vitro data into the models and simulations. So this is just a summary of the talk. We've stated over and over again that botanicals are a complex mixture. 60% of the constituents that we've looked at are neutral, with the remaining 40% divided equally between acids and bases. Most constituents have a reasonable absorption. Agent ginseing's an exception. And then the modeling of ashwagandha indicates that with analytes, good precipitate in the GI tract, and then hydrasting can potentially inhibit 3A4 in the GI tract. So I appreciate your time, and I'm happy to answer any questions if there's time allotted for them. Thank you.
Thanks so much, Michael. We're right up at the time of our break, but I think we can do a few questions just because this is such great work. So from Tim Baker, he asked, how does your data from the compiled model look compared to data from classical studies? In other words, have you validated the model?
No, I haven't taken a look at that for the larger set yet. You know, on the ones we've done with ashwagandha and golden seal, the first approximation is we've overestimated the exposure. And so, you know, as far as toxicity is concerned, if you overestimate it, then you're, you know, indicating that there might be something toxic. It's better to overestimate that than to underestimate that, because if you underestimate that, you might say that you know, it's not reaching toxic levels when potentially it could be in vivo. So that's something we need to do, and that's really kind of the validation of the models, and that's why we want to do more in vivo or simulate in vivo results.
Great, thanks. Question from Dr. Wu. Can the model estimate the concentrations in the brain after crossing the blood-brain barrier?
Yes. You know, as a first approximation, though, we're just looking at passive permeability into the brain, but it certainly can give you an idea of the concentration in the brain. For those type models, you really want to validate those well. There's a lot of transporters that would try and prevent things from going into the CNS. And if you can incorporate that into the model and validate the model with experimental evidence, then you can use that model to ask questions like what would happen if we increase the dose, or other parameters in the model.
Thanks. And then one last question also from Dr. Hu. Can the model be built to take into consideration the impact of food matrix on absorption?
Yeah, so I assume food effects. Yeah, and certainly that's one of the things that GastroPlus is used for in the pharmaceutical industry, is to determine you know, what the exposure of the compound will be if you eat a high-fat meal or if you take it in the fasted state. So yes, you can certainly look at those effects with the models. Now, this is the kind of the high-level GastroPlus PBPK model.
Well, thanks so much, Michael. I really appreciate your talk today. And like you said, this work is ongoing, and we hope to present more of it as we go. So thanks, Michael.
Well, you're welcome. Thank you, everyone.
Okay, so we now have a break until 1:30 in Eastern time, so about a little over 10 minutes. So thanks, everyone. We'll be back in a few minutes. Hi, Vicki. Right now we just see, oh, there we go. So that is the like notes mode. Perfect. Great. I can see you, I cannot hear you.
Can you hear me now?
Yes.
Okay, all right.
Perfect.
Unfortunately, it's at the top of my monitor, so it always looks like it's looking down on me. Okay, so let me see if I can try and make it look like I'm more looking at the camera, somewhat. All right, does that look like I'm kind of looking at it?
Yeah, it's great. It's from above, but it looks good.
Okay, it switched. I had to switch. I'm used to looking at this screen, so now I need to look at this screen so I see what's going on. All right, are we leaving me like this, or should I stop sharing and?
Up to you. You can leave your slides up, and if you want to take video off.
Okay, and then I'll just pop video on when you introduce me, and that way I
Sure.
was just thinking everything will be set and stuff. So perfect.
Perfect.
All right, thank you so much. I appreciate your help.
Thanks, Vicki.
Okay.
Hey everyone. So we're at the end of our allotted time for our break. Vicki, are you there?
Yes, I am.
Great. So our next speaker is Dr. Vicki Sutherland. Vicki is a toxicologist in the Developmental and Reproductive Toxicology Group at the National Toxicology Program. She co-chairs the Botanical Safety Consortium's Developmental and Reproductive Toxicity Technical Working Group. And today she's going to discuss the screening assays selected to be piloted by our various toxicity working group. So thanks so much, Vicki.
Thank you, Connie. Thank you for the introduction. And as Connie said, I'd like to talk to you about the Botanical Safety Consortium and what's been done to include new approach methodologies for botanical assessments. So the outline of my talk today is as follows. I'd like to remind you of the charge for the Botanical Safety Consortium, discuss some of the problem statements, and then show you the current working groups, the assay guidelines, and the strategy details associated with what we've been looking at, as well as the assays that were selected by those work groups and the framework uses. So the current charge for the Botanical Safety Consortium is to evaluate the suitability of alternative assays for botanicals as complex mixtures. And the focus of this presentation is outlining where we are in the process. And this is important because globally, the majority of the testing schemes we utilize to understand toxicity are built on single chemical assessments, not on complex mixtures such as botanicals. Which means that, unfortunately, there's not only little availability for toxicity information on botanicals, but a lack of consensus regarding what comprises sufficient safety for botanicals, and a lack of effective tools and assays to help determine potential toxicities. So where are we now? And what do we want? Where do we want to be? Now we currently don't know all of the assays or the new methodologies that can be used to effectively and reliably evaluate botanicals. So we're working to not only identify which in vitro and in silico assays we want to use and assess, but plan to use and evaluate the application of these tools, which can then integrate the information and tools and approaches to develop a testing strategy and functional framework that allows us to eventually provide information that can then be used to evaluate the safety of botanicals. So to start, I want to bring you back to one of the slides Cara showed this morning and highlight that we've broken these into a number of groups that are working toward this goal. And we've focused them into different themes, toxicity endpoints, exposure and TK, analysis, and the foundational work. And for the toxicity endpoints, we have the teams looking at tools to help evaluate everything from hepatotoxicity, cardiotoxicity, neurotoxicity, genotoxicity, developmental and reproductive, as well as systemic toxicity. So all of the ones with the asterisks next to them are ones we're trying to find assays for. And you'll note this includes all of the groups under the toxicity endpoints, as well as the botanical drug interactions, which is clearly an important issue to address with reliable assays, as we need to understand how botanicals interact with drugs that someone may be taking. And this botanical drug interaction group also aligns nicely with hepatotoxicity under the toxicity endpoints, because there's some nice overlap and interactions across the themes as well. Most of these on the right are more cross-cutting in nature, and not only do they have efforts that span across all of the groups, but they provide the foundation for some of the decision-making, determination of best or potentially overlapping assays, and arguably one of the most crucial parts, analysis and assessment of the assays chosen. So we've established some general guidelines to help each of the groups when looking for assays and methods for the team to test. The assays need to be non-mammalian models. They should be in vitro or in silico models that are already established and reproducible in individual chemicals. They need to be relevant for key mechanisms for botanical induced toxicity. And the assays selected for testing need to be accessible for use by many groups, as well as being suitable for botanicals as complex mixtures, and then available at a reasonable cost. So with these considerations in mind, we've selected assays that essentially break into three areas. And those are the in silico methods to say help with structural alerts, screening assays to define select endpoint toxicities that I mentioned a few slides ago, and potential assays that we can use for mechanistic follow-up to say understand why a botanical might be causing a toxicity or toxicities. And obviously, this last category would be utilized at a later stage in the process. So to remind you of the strategy for this work, I want to show the overarching slide on this and highlight the early efforts, the parallel work stream, and the end goal of developing the framework, and take a moment to point out where we are in the process. And that's under the areas highlighted here, around the chemical and data analysis evaluations and compiling botanical library and list of assays and approaches. And you'll note that, as mentioned earlier, we are working on these in parallel. So for the chemical analyses, we are authenticating and quantifying constituents to be compiled into that botanical library we keep mentioning. And in the parallel work stream, the groups have looked for different assays and approaches that may be suitable for botanicals. And once selected, they will then work with a chemical analysis group for things such as formulation of the product and doses, and with data analysis group, and to best design the studies. So then the question is, how are we going to do this? So here's an example process. For each of the assays, we have appropriate positive and negative chemical controls, and these may or may not be botanical, but they will be positive and negatives that are appropriate for that assay. We will expose the model to the chemicals and the botanicals at select exposures and durations, and measure the endpoints and analyze the data. We will then take all of this information and compare it to the existing toxicity data, including what we found in the literature, the in vivo data, etc., and we'll refine and retest as needed. If this works, we will then integrate the assay into our testing paradigm or framework. So in the next few slides, I will show you a list of the assays we currently have for the screening botanicals for the select toxicity endpoints mentioned before. For genotoxicity screening assays, we have the in silico assays and the models for lead scope, the Ames test for mutagenicity, and the in vitro micronucleus assay. For developmental and reproductive toxicity screening assays, the group is looking at the in silico assays using the PG and DART profiler in the OEC toolbox, transcriptomics in human cell lines to provide information on which can provide information on other endpoints, and both the zebrafish embryo and C. elegans assay. And both of these can also be used for neurotoxicity evaluations as well. And then there's also the DevTox Quick Predict assay. For hepatotoxicity assays, for in silico, we're looking at the Meteor Nexus, Case Ultra, and ADMET Predictor. Transcriptomics in human cell lines again, and these will also help provide information for the botanical drug interactions. And then the NTT cell viability assay, the ATP depletion or mitochondrial dysfunction, and then oxidative distress and necrosis assays, as well as cytotoxic reactive metabolite assays. For neurotoxicity screening assays and the models, they will include the multi-electrode arrays to look at changes in neuronal signaling, as well as the zebrafish and C. elegans I previously mentioned. And for cardiovascular toxicity, we're exploring any of the in silico options and analyses that can be done. We don't necessarily have specific programs identified for in silico work at the moment, but we're looking at a number of the other assays that range from simple cell viability and cardiomyocytes to mitochondrial dysfunction. And one of the things of interest is that some of these assay types, such as the mitochondrial dysfunction, are being used by multiple groups. However, each group may be using different cell types. So while the data can help inform the overall efforts, they are not necessarily a duplication of efforts. However, as I mentioned before, you'll notice that both for DART and neuro, C. elegans and zebrafish would be used. And for these assays, they aren't a duplication of the assay, but evaluations that will include those multiple morphological endpoint assessments, so that each assay can be incorporated into the framework using multiple endpoints. And then last but certainly not least is for systemic toxicity. We plan to use GastroPlus modeling and the human dynamic multiple organ microphysiological system with human tissues. And as I mentioned, the cell types can and or are different for each group, but we have some assays that overlap, hit the same pathways, but with the overarching goal is trying to see what works. So I want to bring you back again to our strategy slide and remind you where we are and that the group has gotten to the point of narrowing down the botanicals and choosing the assays to test. And then where we plan to go over the next year or so, which is to evaluate the botanical library once we've identified the suitable approaches and assays that are applicable, and then evaluate the usefulness of these assays and report the results from the assays and botanicals, and move toward a development of a reliable toolkit and framework for assessing any of the toxicities. And what we're making from and what we are making with this framework, and why are we making this framework? How can it be used? We could screen new and/or data poor botanicals to look for toxicity flags. For the many botanicals already on the market, we could screen and compare them. And we can prioritize botanicals for further testing or deprioritize ones that we believe further assessments are not necessary for. We can also fill knowledge gaps for commonly used botanicals. And we're hoping that anyone could use this framework to evaluate botanical safety, to fill in those data gaps and understand the potential toxicities associated with botanicals. So I hope that I've given you a snapshot of our charge, our goals, and where we are. And we'd be happy to address any questions. Thank you for your time, and thank you very much to Connie and Michelle for the excellent support and the opportunity to present.
All right, I was on double mute. Great, Vicki, thank you so much for that talk. We actually have quite a bit of time for questions, so if there's any questions, please enter them into the chat, and we're happy to answer them. One thing I would like to just say is we are open to hearing about other assays. As Vicki presented, we want them to be established and work well for single chemicals. However, if there is one you would like to suggest, please feel free to email it to us. Okay, just give it a few seconds for any questions that might be coming in.
Connie, I was just also going to say there were a couple other questions that we maybe didn't get to that we could also circle back to from some of the other speakers if I think we have most of the speakers on, so just a suggestion as well.
Sure, we can do that. Let me go back up. I know we had some from Vincent. Vincent, are you still on?
I am.
Great. There was a couple that came in for you. There was one that asked about doing the CCID. Is there a way to determine stereochemistry from Joe Betts?
So that's certainly difficult, right? Isomers still prove to be a pretty big challenge, and not really. I mean, now with the approach we have here, it's more of a general approach. From a toxicological standpoint, if that was an important aspect of a particular molecule, we can dive deeper into that. But the general process of the CCID wouldn't necessarily distinguish that. If they chromatographed separately and we had a standard, that can help, but depending on what type of isomer we're talking about, it may not be.
Thanks, Vincent. I'm looking to see if there's any more for you while we're going through.
I think we covered the ones for
Vincent.
Yeah, I guess there was another one from Ceramia related to, and maybe you covered this, a level of detection for the CAD.
Sure. So yeah, I don't think I mentioned that in the talk. So for the CAD, we get down to, I'll say about 250 to maybe 500 nanograms per mil. So 250, 500 ppb is kind of the range of the detection limits on that.
Thanks, Vince.
There are a couple more coming in about assays right now. So do the cytotoxic metabolite assays include metabolic activation, with glutathione trapping and measurements using mass spectrometry, mass spec, excuse me. I think Amy's on. I might, if she's paying attention, might pull her in to see if she can answer that one. Amy, if you're there, you are now able to speak. For that one, I know it's Dr. Lee, Dr. Albert Lee is proposing that assay. I might have to go back and look to see the specifics. I know glutathione is involved. I'm not sure about the mass spec piece. Michelle, do you remember that?
I do not. I was going to say, I think Amy or Stephen, I don't know if Stephen is on, would probably know that. We might have to get back to you on that, Richard.
All right, and then from Joe Betts, thank you. Nice to see assays for cardiac cell conductivity endpoints. Ephedra seems to exert bad effects by inducing arrhythmias. Yeah, that's exactly right. Hopefully we are able to capture that in the assay since that's a known issue with ephedra. Question from Bill, have you used MTS assays for any of your cellular toxicity testing? I think MTS versus MTT came up for the hepatotox group. They had a rationale for why to use MTT over the other. I have to go back in my notes and see why. It's not at the exclusion of MTS though. We could see if that one would work better or would work as well as some of the other cell viabilities that are proposed or other assays proposed.
I think they're relatively similar.
Mm-hmm. Okay, I'm just trying to look through as we go. If there's other questions, please feel free to enter. We're a good bit ahead of schedule and our next speaker isn't on yet, so we have some more time for questions here.
There was one other comment related to Michael's presentation on the golden seal. There was one question related to the hydrastinine level of the starting material. And I know I did not see it in the paper, but that's something that hopefully we can follow up on. And I know several of the authors of that paper are involved in the consortium. And then I did also just want to mention Bill Gurley had put a note in that most golden seal products recommend a dosing schedule of 3 times per day. So that CYP3A4 inhibition that dose could be reached using based on that schedule. So really nice to see the modeling of that work.
Agreed. And I'll just make one comment. I think Michael might have said it, but for the modeling and the constituents he showed, those are ones we found from existing literature. And as we go through with some of the work Vincent's doing, we can add to the list of constituents we have for ashwagandha and others and try and get the amounts as well to try and feed those into the models.
Hi, Connie.
So Connie.
Oh, sorry. This is Cynthia. I just wanted to go back to a couple of questions that were asked of Cara's talk. Someone mentioned, why didn't we select vegetables or fruits or something in the food stream as negative controls? And just wanted to point out that we were aware of some work using extracts from common foods in high throughput screening. So we have that paper, and we do want to use that as a comparator wherever possible in the future, looking to the future. And I had another question that came up about whether we were using extracts versus finished versus raw material, and I just wanted to make sure that we were clear that we're using extracts typically, not the raw material, not the raw botanical material. Thanks.
Yeah, thanks for those clarifications, Cynthia. Someone else was about to speak. Was that you, Holly?
I think it was Vicki.
Oh, sorry, was it Vicki?
Yeah, I was just going to ask, it just dawned on me that at least going through grad school, the majority of our work that was necessarily negative or didn't produce anything exciting wasn't necessarily published. And I was wondering whether or not HESI has a way for people to kind of share anything that might be helpful in that way. Meaning, you know, if you've done some work on some of these botanicals, but nothing showed up, you know, if they'd be willing to share what assays they did and the information associated with that.
Yeah, I think that's a great idea. I mean, I think there's, you know, we would be very, very open to seeing any of that information. Like you said, a lot of times that isn't what's published. So if there are people on this call or people that you're aware of who have done some work, especially whether it's in vivo or with any in vitro assays on these, and have found negative results, but maybe haven't published them, I think we can create a mechanism to have that information shared, you know, through part of the consortium. I know we would definitely be interested in using that as we work on the literature reviews for these botanicals as well. But I'm open, Vicki, to suggestions on how we can better facilitate that, because I do think that's an important point.
I wasn't necessarily sure, but I thought we could throw that question to the people on the audience today, that they might have some suggestions or at least be willing to reach out to you to let you know if they had some information, to see if it was worth putting something up so that people could help participate in the efforts.
Yeah, we can definitely put a call for that up, and then depending on sort of the input we get, create something more formal. But I think for now, having that as kind of an action from this is a great suggestion.
I thought of one more question that I wanted to circle back to. There was a question about testing one version of each of the botanical ingredients versus looking across different variations in the marketplace. And I just wanted to point out that there are some parallel projects with different ingredients that hopefully we will get into that in future iterations as research projects, to look across multiple versions, and point out that some of our members like Nadja Czech has some amazing work looking at, you know, 50 different versions of kratom. And I know at NTP we've done some work looking at sufficient similarity of complex botanicals, including things like ginkgo and black cohosh and echinacea. So we are, you know, focused on that as a future iteration.
Thanks, Cynthia.
Yeah, a couple more questions are coming in now. So one from Annie. So she asked, or they asked, a lot of plants that we have chosen to start with are imported. Is there any prospects of analyzing flora that is local to North America, in the spirit of respecting our ecosystems and valorizing them? So that wasn't really a consideration we had when choosing. It was more looking at the existing toxicity or lack thereof information. I think it's something that once we have a toolkit or a few assays working, we could definitely look into testing some flora from the North America or more local that maybe doesn't have as much existing information or would be of interest. Okay, there's another question, or maybe a comment from Joe Betts that says, there's some bioactives in conventional foods that might be considered toxins, certain ones in carrots and rape seeds, and different ones like broccoli. He listed a couple examples. The dose makes the poisons though, so that's a really good point. And Cynthia, I think that was highlighted in that paper you were referring to, right? That even though they were food products, there were some maybe signals.
Exactly.
Yeah, sorry.
Yes. Yeah, and I think part of our challenge, as we like to reiterate, is determining the difference between seeing activity and seeing adverse activity in the different bioassays. So I think that's a really important point that we need to keep in mind and focus on moving forward.
Thanks, Cynthia. And just a comment from Richard Van Bremen, why not organize a special issue of a journal to focus on safety studies that show no toxicity? And that's a really great idea. And as the information comes out, if we could do that, I think that would be great. All right, so we're a few minutes ahead of schedule, but I think that's all right. And we have our second keynote that is going to go on next. I'm going to turn it over to Holly Johnson one of our steering committee members to introduce Dr. Lodog.
Great. Hello, everyone. This is Holly Johnson. I'm the Chief Science Officer for the American Herbal Products Association, and happy to be here today and be a part of the Botanical Safety Consortium. I really could not be more pleased to introduce today our keynote speaker, Tieraona Lodog. Dr. Lodog is an integrative physician and a leading expert in safe clinical use of botanical medicines. She's formerly the Fellowship Director at the University of Arizona Center for Nurse Practitioners. President Bill Clinton appointed her to the White House Commission of Complementary and Alternative Medicine Policy, and she then went on to serve in the Advisory Council for the National Center for Complementary and Alternative Medicine, and has chaired numerous dietary supplement and botanical panels at the United States Pharmacopoeia. Over her 4-decade career, Dr. Lodog has received many awards from academia, public health, and industry. She's really a legend. She's an incredibly powerful human being. And with that, I could not be more pleased to introduce for what will be a wonderful talk, Dr. Tieraona Lodog.
Hi, am I going to be sharing my screen, or do you all have the presentation? Can you just tell me how you want me to do that?
Either way, I made you a presenter, so you can feel free to, or I can pull up your slides now.
Go ahead and pull up my slides.
Perfect.
All right.
Let me just get a little organized. Hopefully you all can see that now. Does that look okay?
There we go. That's perfect. Thank you. So you'll drive for me.
Yes.
I am so happy to be here with everybody today. Thank you for the invitation. When I was asked to present, I really wanted to give a perspective of a clinician in practice and what it's like to interface with dietary supplements in general, but botanicals in particular. So I want to disclose that I have used herbal medicines in practice for 40 years now, a long time. So I feel comfortable with them. However, we are facing challenges today with the just vast number of products and the polypharmacy that our patients are taking that it's very difficult for clinicians in the field with 20-minute visits to really have a conversation and help guide patients the way we would like. So this is a real world take on botanical safety. Next slide. So that is just kind of the look, right? You go to any store, my children don't let me into Whole Foods or, you know, any place anymore where there's a supplement aisle because they're like, Mom, keep moving, keep moving. Do not look down that aisle and don't go talk to anybody. Because, you know, what's really common is how many people are sort of standing there looking at an aisle and asking usually a very pleasant person, I have diabetes and high blood pressure, what should I take? And there's this myriad of products. And for me, I'm always like, you know, are you on medication? What kind of blood pressure are we talking about? You know, all these kinds of things, which you can't get answered in a supplement aisle, but this is where people are going to get their information. And the sheer number of products out there can make even somebody who has a lot of expertise in herbal medicine overwhelmed. Next slide. So if I was going to summarize the challenges that most clinicians face, we're concerned over quality because we hear about adulterated products, contaminated products. FDA, you can go to their website where you can see the adulterated products, usually sexual enhancing products for sex, for sexual dysfunction, erectile dysfunction, or weight loss. You know, those are kind of the big categories, the Sodom and Gomorrah of our supplements. But I think overall clinicians are just kind of nervous because they don't usually hear about the companies that are doing it right. They hear about things when they go wrong. Herb-drug interactions, you cannot look historically, not really. You can't look for the most part at a plant that was used hundreds and hundreds of years ago for something and predict if it's going to have an interaction with a specific drug and drug metabolizing enzymes. And because so many patients have multiple morbidities, many of them are on way too many medications. I mean, a lot of people just take 4, 5, 6, 8 drugs, and then you layer on top botanicals or other dietary supplements. And it's very hard for us to have a really good predictive model of should we be concerned or not. And unfortunately, what that usually means is that clinicians err on the side of caution and just say you shouldn't take it, right? Don't take anything like that. Or worse, they say it doesn't work and kind of invalidates the person's value system or their desire to use things that they perceive as more natural. The dose. What is the dose? What is the dose for a tea? What is the dose for just ground up herbs in a capsule? What is the dose for an extract? What kind of extract? You know, what is the safety profile for a 3-year-old, for a pregnant woman, for a 75-year-old, maybe with some diminished renal function? I mean, so safety is not just does it cause cancer long-term or genotoxicity, it's much broader than that because of the vast spectrum of people we care for. And what are the anticipated adverse events? So what should I be looking for? What could I even counsel my patient about what we think the adverse effects might be? It's unclear to where to go for unbiased and really clinically relevant information. I'm going to share a resource that many of us use a little bit later, but I think this is also like, just where do I go get the information that doesn't just say echinacea is, right? Echinacea root or echinacea herbal extract does. We need more specificity to this, and I think this is one of our challenges. And then the problem with the way the market is and the way we train clinicians is that without the proper training or tools, it's hard for us to make recommendations to patients who bring things in for us to see. And it's also difficult for us to make recommendations for a botanical that we think actually might help this patient. So it goes two ways, one for mitigating risk and the other for giving something that may be safer for a condition for which conventional medicine does not have great treatments for, but not feeling comfortable or confident how to make those recommendations. So it's kind of on all sides. Next slide. So this is just an example of a patient I saw recently, a 62-year-old man, type 2 diabetic, elder Hispanic. He has high blood pressure, dyslipidemia. He has reflux from kind of gastroparesis from the diabetes. He has back pain and arthritis of his knees, in no small part because he's also heavy. He's got a BMI of 30. He's taking metformin, an ACE inhibitor, a statin, a PPI, and he takes acetaminophen for his pain. And he comes in for a 20-minute visit. That's all we've got scheduled to go over his labs and to discuss where he's doing, refill prescriptions, etc. And he says that his back and knee pain has gotten worse and he's wanting to use something natural, and he's brought in 2 products with him and he wants to know if it's okay to take them, right? So, you know, this is the whole thing. It's like you bring in these bottles. If it was one ingredient like turmeric or something, it's a lot easier to deal with, but many patients bring in complex mixtures and you're trying to decipher that in your visit. Okay, so if we go to the next slide, I'll show you what the label showed. No product names, just the labels. So you can see there were 2 products. One is he's supposed to take 2 capsules twice a day. It's got a white willow bark extract, and it tells you how much salicin is in it, 60 milligrams per 2, so he'd be getting 120 milligrams of salicin per day. A devil's claw extract doesn't give you the extract ratio, but you know, it does actually. That one shows you the extract ratio, but a lot of clinicians don't understand actually what that means. Then you have a theracumin or a curcumin at an extract, which actually is a lot lower potency than what most clinicians would probably be used to seeing, right? 60 milligrams. Boswellia extract, it's standardized to boswellic acid. You can see how much you'd be getting, 180 milligrams per day. And then it's got this blend, this polyphenol blend of 100 milligrams of blueberries and raspberries and, you know, not that those things are bad for you, but you'd be much better off eating a half a cup of blueberries or something instead of getting 100 milligrams of these powders. Now, if you look to, so I actually think that label is pretty good. I think that label is easy to understand if you understand about extracts and marker compounds, or it's done well. I want you to move over to the right to the other product the patient brought in, which this is a popular product actually in our area of New Mexico. And it's a proprietary herbal blend. So it's got 2.6 grams of this combination of herbs. And you can tell from the plants that are in it: cramp bark, Jamaican dogwood, black haw, kava, passiflora, valerian. I mean, you can see by looking at these, this is sort of joint and muscle, so muscle relaxation. So he was recommended these two products by somebody working at the shop, and this is what he brings in. So that was to take 4 capsules 2 to 3 times per day, and he made a big deal of letting me know that when he went and looked that up, it is made in a US FDA regulated facility, which to him, this elder Hispanic man, what he thought that meant, it was kind of like it's a regulated FDA product, right? So it kind of had a little bit of in his head, not quite understanding what that meant, right, for GMPs or anything like that. If you're not a part of this industry, you're just a consumer, that's what it meant. So given what we know about the medications that he's taking, his underlying problems, his age, I have like 8 minutes to look at this and tell him if he should take it, not take it, and try to do it without just saying, no, you shouldn't take it because I don't know if it's going to interact, or I don't think these are good products, but to actually be thoughtful. I want you to think about a clinician who knows nothing about herbs, other than what most consumers know, that turmeric can be good for your arthritis, or, you know, they've heard of willow bark because that's where aspirin came from. So most clinicians don't have a depth of expertise in this area. And so this is what they're confronted with, these 2 bottles of somebody who comes in on polypharmacy and who has multiple problems, health problems. Next slide. So if we think of willow bark, just like think of a couple of these things, and if you did a quick search, right, like what you might find. We know that willow bark, not surprisingly, can be effective for back pain and arthritis. Why would it not be? Because, you know, it has significant anti-inflammatory effects, right? We did a review with our USP panel, Dr. Helen O'Ketch and Robin Marles and Scott Jordan. They let me participate in the writing of this article. But we did a review on willow bark, and what was really interesting is that on low dose aspirin that contains 62 milligrams of salicylic acid to 81 milligrams in that range, we require a warning on it about pregnant women, use in young children, impact on bleeding, etc. But when you look at willow bark supplements, many of them deliver, you know, up to 240 milligrams of salicin, which can be metabolized to create 113 milligrams of salicylic acid. So this product contains 60 milligrams of salicin per serving. So in a day, we'd get 120 milligrams. So more than what you'd be getting in a low potency aspirin. Now, for me, I wouldn't be overly worried in this patient because he's on a proton pump inhibitor. But what if he wasn't on a proton pump inhibitor? Would this be safe long-term for him, given the fact that we know that low-dose aspirin is significantly associated with GI bleeds? It's not just full-dose, full-strength aspirin. So, you know, our USP panel in our publication recommended that there should be some sort of labeling for this to help guide consumers more effectively. But again, so yeah, I would think this would work. I probably wouldn't worry in this patient because of his PPI use, but what if he wasn't on one? What is the safety long-term, and how different is that from just a small baby aspirin? Next slide. When it comes to devil's claw, a plant that comes to us from Africa, the southern and eastern regions of Africa, This is a plant that has a long history as an anti-inflammatory and an analgesic, so it's been pretty well established. There's a lot of studies that confirm that it has beneficial effects in back pain and arthritis. So this is what he's got. He's got back pain and he's got osteoarthritis of the knee. If you look at the safety reviews, that they found few adverse effects compared to placebo, with one exception, and that's loose stools, right? Up to 8% of patients report diarrhea, especially when they get into the higher dosing. So, you know, I not much other information out there with this. It could be efficacious, don't have a lot of data on drug interactions, but the safety data from 28 studies showed it was pretty well tolerated without significant adverse effects, except some GI effects that were more than placebo. Next slide. So then I'm moving along and we come to turmeric. And I think most clinicians are really interested in turmeric. A lot of conferences I go, people have a lot of questions. Doctors feel like there's something to it and that it works. There was a Tufts review that actually found that curcumin was more effective than placebo and was pretty similar for NSAIDs for pain relief and for functional improvement, specifically in knee arthritis, which is what this gentleman has. I think that one of the big questions becomes then, how much do you need? What's the dose? And does it need to be like taken as like with a phytosome? Does it need to have piperine in it, you know, the black pepper alkaloid to make it more absorbed? Or like some of us believe, maybe a lot of what it's doing is actually in the gut itself, decreasing intestinal permeability, reducing inflammation, blah, 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 blah. And you may not need this kind of big absorption. So it's generally 1,000 to 1,200 milligrams per day of extracts that are standardized to the curcuminoids. This had 60 milligrams. It is supposed to be a more highly absorbed curcumin, but this is also what I mean. It makes it hard. A clinician's looking at that and isn't sure. It's like, well, is this enough? It seems pretty low. Pretty low probably won't harm you, but pretty low may not help. Or do they know about these other types of turmerics that have purportedly a better absorption and bioavailability? Next slide. So he was also in this product was Boswellia. These are often found together. That Tufts review actually included 4 randomized controlled trials that looked at Boswellia, standardized extracts of Boswellia, and it did show efficacy, right? This is a resin, an oleoresin, a gummy resin from the Boswellia tree, also known as frankincense. The safety up to 6 months at doses up to 1,000 milligrams per day appeared to be well tolerated in the reviews that have been done. I think it's important to note, not that it doesn't mean that it's not, that the randomized controlled trials aren't excellent. It's just 75% or 3 out of 4 of the studies were done by the same manufacturer. So you always like that independent research when you can get to it. For me, Boswellia definitely has anti-inflammatory effects, and a lot of times we use it in respiratory problems because of its effects on 5-LOX, which tends to be more indicated for sort of bronchial problems. But Boswellia is used and we don't see a safety issue. So I'm thinking, okay, this product, I'm feeling pretty good so far. Next slide. So then I look at this other proprietary herbal blend. I didn't go through all of them, just a few of them. But, you know, one that is interesting because herbalists really like Jamaican dogwood root bark. So it's a large tropical, subtropical tree. The bark is removed from the root. You can tell, for those of you who maybe remember some of your Latin and Greek, that Pisidia actually means to kill fish, right? That's kind of the origin of that word because it's used as a fish poison. Hence its name is for this. And that's because it contains rotenone, which is toxic to fish. And this is one of its historical uses. I think that issues around safety are simply that we know that that's a neurotoxic pesticide, and it's been found in streams and water, and there's a question about its linkage to Parkinson's-like symptoms, and they're even using rotenone to induce a Parkinson-like model so that they can test treatments. So what is the safety? We don't know. What are the clinical trials? Really don't exist. history of use as an analgesic and sedative, but what about chronic daily use of it, and at what dose? Joe Betts, I heard earlier, said the dose determines the poison. It's again, like, is this something we should be concerned about? But if you're a doctor and you go look that up and you see something like that, it would make you very hesitant to be supportive of this product. Next slide. Lobelia is also in it. Again, a lot of times used as a bronchodilator, but it is also used as an antispasmodic. Topically, it's used a lot for aches and pains and joint things. Lobelia, is an interesting plant. I remember once at my herb school in Albuquerque, I shared the school with an herbalist named Michael Moore, who was just a character and a brilliant, brilliant fellow. And I'd gone in, and he had come back from his harvesting. He would take lots of students out, and they would go harvest and come back, and they'd be tincturing and making things. And so I sat down, I was chatting with him and his partner, Donna, and there was tincture right there. It was lobelia. So anyway, I just took some and dropped some in my mouth. And Michael at that time goes, no, or something like that. I'm like, what? And it was lobelia. I just took a little bit. Well, it was fresh lobelia. And I never felt sick to my stomach, but I was really, really altered for about maybe 20 to 30 minutes. I felt very strange. It was very uncomfortable. I'd mostly used dried lobelia, so I was not really familiar with fresh, which was very potent. It is known as pukeweed. It was used a lot by indigenous peoples to induce vomiting before ceremonial types of events where vomiting was part of it, to cleanse you, to get you ready for the ceremony. So it is a potent emetic, and we have, there's been this huge debate about the safety of lobelia because many herbalists would argue that you vomit before any real toxicity like respiratory depression or anything like this could happen. It is a partial nicotinic agonist, so it is potent. The FDA listed it as an unsafe herb, and that was back in the 1980s, really, when it was being promoted heavily to help people stop smoking. So again, if you're a clinician and you're just kind of looking it up and it says somewhere that FDA says it's unsafe, that's going to be kind of just the end of the deal for most clinicians. They're going to look at this and say, no, don't take it. Next. And it had St. John's wort in it, which some of you may think, well, that's an odd herb to have in there. Well, it's actually not. A lot of traditional herbalists use it for kind of nervy pain. It's used topically and internally for that purpose. Of course, most of the data on it is for depression. I would contend as a clinician that lots of patients who have chronic pain are also depressed. So the inclusion of it kind of makes sense. The adverse events are pretty comparable to placebo, though I will say, living at 7,500 feet above sea level, we do see photosensitivity with people who are taking St. John's wort, and had a lady once that went into a tanning bed while she was taking quite a bit of St. John's wort and had a very nice burn. So photosensitivity can occur just like it can with other medications. I think the big thing is, specifically, he's on 2 medications that could interact with it, right? Based on human pharmacokinetic studies, his PPI and his statin. So everybody's aware of the impact that hypericum has on some of the CYP enzymes as well as P-glycoprotein. So that's in there, and that would be one that doctors would see. Even if they didn't know it was specifically for these drugs, they would have a sense that there may be a problem. Next slide. So when we look at this herb-drug interaction, like with St. John's wort, I have to say, and I tell clinicians this too, is that there are some herb-drug interactions. There's no question. And they're not as many as predicted by the preclinical research. Preclinical research always shows that there's, that it interacts with everything. But when you actually give it to real human volunteers, preferably with some genetic testing also to know if they have any genetic polymorphisms in their enzymes, drug metabolizing enzymes, we see that there's just not that much. And this could be, I mean, intestinal transporters are one thing, but for some of these enzymes, you just may not get enough blood level. You may not get enough rise in the serum level to really cause an interaction. Next slide. There was a great article that I refer to. This is just one little section of it that actually looked at the interactions, and you can see on the left there's this preclinical, and it looks like everything interacts with everything. I mean, it's just if you see the ginkgo, all the enzymes, ginseng, golden seal, green tea, isoflavones. But then you look over to the clinical interactions, inhibition and induction, and here you actually just don't see much effect, with an exception for golden seal. Golden seal, Oregon grapefruit, barberry, a number of these contain pretty potent alkaloids, berberine, for example. We know that actually that can be a pretty significant inhibitor of CYP2D6 and CYP3A4, which between the two of those constitute, what, 60–70% of all drugs. It's heavily promoted for diabetes. It's heavily promoted for cardiovascular problems, and the data is pretty compelling. However, most of these patients that are using this are also taking these medications. Golden seal, Bill Gurley's work there did show this interaction. So, but if you look at prediction here, preclinical versus clinical, big difference. Next slide. When we are looking at herb-drug interactions, many of us do use this database. There are pros and cons to every database. This is one that's used. You can see on the left side, interaction checker, effectiveness checker, pregnancy, lactation, adverse events. So a lot of clinicians feel like if there's only one place I can go, this may be the place to do it. Next slide. So if what it allows you to do is put in all of the drugs and all of the botanicals, and then it ranks them by red, yellow, or green. Red meaning potential for serious interaction, yellow moderate, and then green really minor, and I would say mostly theoretical. But you could see that if you're a clinician and you had the time to go do this, log in, get here, put all these in, and it spews out a whole bunch of moderate interactions and a couple of reds, it's going to give you real pause. But that would take a while to do this, right? To actually go in, enter it all, and then print it out. Next slide. But it is a good tool. My point of this was simply to say that the dynamic between a clinician and a patient is complex, and that a lot of us in practice, in primary care practice, we see people who have a lot of different problems, health problems, and they're on multiple medications. And when it comes to safety, it's not just will it interact, but if I have a patient with diminished renal function, what's that going to do? You know, their creatinine starting to creep up. I've got a patient taking a hepatotoxic acetaminophen, 3–4 grams a day for pain. And what happens if you layer kava, which was in that one product? So I think that, when we're looking at tools, we're going to have to find and research. I think we're going to have to just up the game when it comes to making labels more transparent for people to understand, having dose ranges that are more meaningful, more research on how these some of these herbs are metabolized and excreted. And I would also say that adverse events are not common. Serious adverse reactions are not common. They're not, but they do happen. They will happen. And hopefully you can just maybe be a little sensitive to busy physicians who are seeing 20–25 patients a day, and people come in 4 or 5 every day with a bag full of ingredients and products, and just how complex this situation is for clinicians to try to use good judgment. And unfortunately, the answer is often just don't take the product, which is not the right answer either, because some of these might really help. I would have told the patient, and I did, that I thought the first product was fine for him to use. Let's try that. And that I wasn't comfortable with the second product with some of the ingredients in it. And so let's see what happens if we use this one and give it 6 to 8 weeks. Let's see how it's doing. Get you some water therapy, some PT, some other things. But I hope that I gave you a little glimpse into the world of what clinicians are thinking when they're having to think about safety in the clinical setting in usually a relatively short time frame. Thank you.
Thank you so much, Dr. Lodog. That was a fantastic presentation. We have a couple questions that came in. So one from Joe Betts. Salicylic acid is highly bound to serum albumin and will displace albumin bound to active pharmaceutical ingredients. I think that was just a comment.
Yeah. Joe knows his herbs.
Okay, another question for Dr. Lodog. I learned that natural salicylate is, excuse me, I probably said that wrong, but cannot be equated to aspirin, and acetyl function on the molecule, making a large difference on the medicinal effects. Have you observed something in that sense in your clinic?
I have, a lot of patients don't use much willow bark. You'll see them in products like this, but willow print and some of these other things were really big a number of years ago. And I got to be really honest, most of the people I see are taking things like turmeric. It's the _1 in practice that you'll just see. When we did our review on willow bark, a lot of this depends upon gut microbiota because, the precursor is actually, what's in the plant, and then this is activated and converted by gut microbes into an active form. And, I think when we did our USP review, I'd always heard that, willow bark, you didn't have to worry about the blood, coagulation. Didn't have to worry about Ray's syndrome in children. I'm not sure that that's actually accurate. I'm not sure that that's completely accurate. The acetyl group definitely changes it from what is found in the plant. But the salicylic acid itself that is generated has these properties to it.
Great, thank you. I'm just looking through. Joe Betts had another comment that electic physicians derogatively referred to lobelia doctors because of their propensity to poison people.
I know. It was always the battle between the eclectics and the Thomsonians, right? Thomson was kind of this big believer in lobelia, and so you had the Thomsonians that kind of all use the lobelia, and then you had like the Kings and Felter and the eclectic physicians who really felt that lobelia, definitely could be pretty toxic. Yeah, these old divides are quite interesting, but that was the Thomsonian versus the eclectics back in the day.
Connie, I think there was I think it was related to this, a comment in the chat from Amy.
Yes.
You see that? Okay.
I just sent her a note to ask if I could read that out loud, but I think it's all right. And she had brought it up within the consortium before. So Amy said, with regards to the Sprouse article and the data table, which we have Richard Van Bremen on the consortium, he was on the call earlier. But one thing to keep in mind is that the in vitro data reported was all from simplistic in vitro models, not cell-based physiologically relevant models. And she commented, if the whole cell models were used.
Yep.
What was that?
Yeah.
Yep.
I would agree, or in silico, or other kinds of other methods. But you have to remember that, yes, but I'm also talking from a clinician's perspective. So a clinician is just sort of looking at all of this, and everything they've heard is ginkgo interacts with everything. Ginseng interacts with everything. You can't use it if you have high blood pressure. You can't, there's just a million things that clinicians have heard. And one of the points of that article was simply to say that much of this research that was initially done in the 1980s and the 1990s that predicted all of these interactions we're done with simplistic and these sort of more primitive or simplistic tools. But that's what generated a lot of the interaction data that is still in books today. It's still what people talk about, and it's still referenced in clinical, in scientific reviews and papers. So it is important to understand that when we actually look at real data, in whether it's more sophisticated preclinical data or kind of the gold standard FDA, sort of using FDA approved probe drugs and looking at in human beings, that they don't match up. And if you looked at the original data looking at interactions, if you were a clinician, you'd tell people never to take any of these because it looks like it interacts with every drug ever made. So again, from a clinician's perspective, we're asking doctors in the field to not only know their own medicine and diagnostics and care for a lot of people with multiple morbidities, but we're also saying, if before you take this supplement, go talk to your doctor. Before you take this supplement, go speak to your clinician. And clinicians are overwhelmed by this information. Just even reading a label with extracts versus not, a 4-to-1 extract, what does that mean? Even something as simple as recommending ginger to a pregnant woman for morning sickness. At least half the products that are in the marketplace are very concentrated ginger extracts. They're not just ginger powder. And we know that high doses of ginger are not safe in pregnancies. So here we've got the American College of OBGYN saying first line therapy for morning sickness is to stop your prenatal.
I'm having trouble hearing you.
Is to stop your prenatal and to take 250 milligrams of ginger 4 times a day. So I tell my patient, go find ginger and take 250 milligrams 4 times a day. A lot of these products that contain 200 to 300 milligrams of ginger are 10-to-1 extracts. If you take 4 of those a day, you're way over 1,000 milligrams of dried ginger powder. So this is what I mean. My talk today was really just from the perspective of a physician in practice who actually is pretty versed in herbal medicine and the complexities that we deal with every single day. And without better tools, without better labeling, without better dosing information, I think I'm grateful we don't see more serious adverse effects, but we will. I mean, you will as people take more and more medications and supplements become more and more popular. So, but I would agree with Amy, but that kind of old information is what's still floating around in there, and you can find it in any paper today on a review of the Botanical.
Sure. And just the end of her point was saying in the BSc, we're trying to focus on some more of those complex models too. Great.
Absolutely.
Question from Minjung Chen says, do you think herbal dietary supplements should be regulated by FDA or other agencies?
I think we need a better model. We need a better model. It's like golden seal is not the same thing as vitamin D. They're just not the same thing. These are complex mixtures. And I think you have to be smart about it. Is cranberry a beverage? Is it an herbal dietary supplement? I mean, it's like there's a lot of overlap here. But, trying to find a model that allows a better dosage ranging, more specificity around the extracts, in a way that doesn't infringe people's access to supplements, but makes it a better model. It'd be hard as a clinician for me to argue against that. It'd just be hard to argue against it.
Great. And the last thing I see, one just came in. How can clinicians actually best inform themselves about herb-drug interactions?
Well, it's using tools like the one I showed you, which is not perfect. It's not perfect by any stretch of the means, but it does help clinicians be able to document in a chart that they did it, and they can print it and put it in there. And for a lot of the clinicians that I teach, I tell them, look, you have to document that you had a conversation with the patient, that you had a conversation, and that you use this tool to help you be able to make a clinical decision about taking it, not taking it, dosage, etc. And right now, I'm just telling you the data, until there's like a database that really pharmacists and clinicians can just go to, that has better modeling and is much more definitive, it's going to be hard. I can rely on the pharmacist, and I do, because if I prescribe a drug to a patient, they take it down to the pharmacy in Santa Fe, the pharmacist will call and say, hey, they're on this, are you sure this dosage range, there can be an interaction. And two parts of that. One is that there's nobody really, not like a pharmacist, that I can just send that's going to be able to really be watching, especially for these complex mixtures. And two, even with the drugs, it's becoming a problem, because a lot of our insurance companies don't want people to go to the local pharmacy. They want them to buy them online, which also just takes one more layer away from somebody who could have contact with a professional, like a pharmacist, who's able to counsel more around drugs. I actually think pharmacists need a lot more training in herbs. They're the keeper of the drugs. And if we worked more on training pharmacists, they could be just the most natural resource for clinicians to refer to. So dietitians are good for vitamins, minerals, omega-3s, and some of these, the more of the micronutrients and that. As far as the botanicals go, I would love to see a more pharmacist education. Clinicians, it's good for them to know, but I'm just telling you, it's a lot. When people come in taking 13, 15, 18 supplements, which is common, not rare, the chance of an interaction is almost 100%. I mean, Goodman and Gilman, remember? By the time somebody gets to 8 drugs, you're like at greater than 80% chance that there's going to be an interaction between them. So just fascinating. So the natural database is what I encourage clinicians to use, that kind of tool, and document it in the chart for medical legal reasons. And I certainly teach pharmacists and I teach physicians, and I think pharmacists need to really take on more of this as they used to. So.
Great, thank you. There's a few more that have come in. One might be good for the open discussion, but I'll field it to you first. Could botanical supplements be divided into high risk and low risk categories?
Absolutely. And high risk, low risk would also, you have to figure out drug interaction. St. John's wort is extremely well tolerated and very safe. It's low risk. It has a high risk of interaction, right? So you'll have to separate those out. But like in traditional Chinese medicine, they have superior and inferior herbs that in their model, and superior are the ones that are more safe, can be taken long term. We don't worry too much about adverse effects from them. And the inferior means that they have the potential to be more toxic and more problematic. That's an interesting model to look at. I think herb-drug interactions are kind of a separate thing, and I think pregnancy and lactation are kind of a separate thing as well. But I definitely think you can rank those. Maybe it's more of a chart with multiple squares, so that just adverse effect and safety, genotoxicity, all these kinds of things, you could have low risk, but then you could have, if it was potential for a high risk for drug interaction. You have to take that into account, because so many people, we just prescribe too many drugs in this country.
Okay, another question that came in says, how does one know that the ingredients and levels listed on the label are actually correct or a good approximation?
You don't. I mean, there are a number of really good companies. So I want to also say this, there are very good companies out there that do a great job. And I hate for them to all be painted with the same brush of irresponsible companies that are out there. But this is always one of the big worries that I think clinicians have. It's like, so does it have 81 milligrams of acetylsalicylic acid, or does it have 106, or does it have 29? I mean, we sort of feel more comfortable with a drug on how much is in it, and that it is a pure compound. by their very nature, botanicals are complex. And so, you know, we should be able to get the milligrams right. But did they get the right plant part? Was it harvested in the right time? You know, was it the ginkgo that was great quality, or was it the little dust that was left in the corner that was picked up and used? I mean, so I think companies have done, a lot of our big companies do a really good job with this. But this is a huge area that clinicians worry about. I'm going to be really honest with you. If you're with 300 docs, they will tell you what are your biggest barriers? I've asked them in conferences, what are your biggest barriers to use? Many of them are very concerned around quality. They're concerned around herb drug interactions. And then 3, just like, I just don't know, can it be used like for weeks or longer? I'm on an oncology chat group, and the biggest concerns for oncologists are patients taking botanicals. It's like, I don't know if it's going to interact with the chemotherapy. I don't know. And so, you know, where some things may actually be protective or beneficial, they're just unwilling to kind of look at it, because it's just, they just feel like, I just don't want to be responsible. So it's a big question around quality, though personally, I think there are really good companies out there that do a really good job. So.
Great. And I think one last question for you. When there are many herbs mixed in one formulation, how do you address herb-herb interaction? Will they have synergistic effects or toxicity?
It depends upon the blend. I mean, so sometimes, like, you know, if you're going to use a laxative, right, something, you know, cascara, senna, a lot of times ginger would be added, right, because it reduces spasms and cramps. So that would be a synergistic effect. You could see that in some countries like in traditional Ayurveda, turmeric and boswellia were often combined, right? So a lot of times there can be synergism, or there could be a corrective measure, right? You've included an herb as a corrective. But the other thing I would say is that a lot of these formulas are just like, we threw everything in but the kitchen sink, right? So we're going to put clove in it, St. John's wort in it, cramp bark in it, valerian, passion flower, Jamaican dogwood, red raspberry leaf. I mean, you know, we're going to put all of these things in there, and it's you know, it's hard to know. Sometimes you can look at the formula and go, I understand why they blended these, and sometimes you look at the formula and go, I have no idea why they did this. But the concept of synergism is, you know, is very old in herbal medicine. The combining of plants is common and is the norm in some systems like traditional Chinese medicine and Ayurveda, and to a lesser degree in Western herbalism, but it still exists. And we've been adopting that too with oncology, using multiple chemotherapeutic drugs. HIV, our big breakthroughs came through when we began to combine drugs together. So we know that combinations can, if they're thoughtful and done well, can often, you know, lead to better outcomes. But some companies, you're looking at their product and going, I have no clue. Women's formulas, particular. Let's throw every female herb in there. Like, let's put everything in there that we've ever heard was good for women, and we'll throw it in there, and we'll give it some great name, and we'll market the heck out of it. And you're looking at it going, I don't even understand I don't even understand this formula. So I think it varies. Some formulations make a lot of sense, and they may be based on tradition. Some are based on science and research that have been studied. These combinations have been studied, and in other cases, they're just kind of not very well thought out and don't make much sense. So,
thank you so much, Dr. Lodog, for a fantastic talk.
Yeah. Well, thank you. Thanks for the Botanical Safety Consortium. Amazing work that everybody's doing, and for all the people on here today that are interested in this. I think that botanicals are one of the most interesting parts of the whole supplement space. And I think it's because I do believe that they have a lot of therapeutic benefits that that some of them, like what would be in the TCM inferior herbs, some of them are quite effective and quite potent, but that also means that they need to have more care and more safety data for them. So I'm excited for what the consortium is doing, and keep doing it. It makes it better for all of us.
Thank you so much for those kind words. And thank you to all our speakers
you.
today. I'm going to turn it over to Michelle for a few updates, and then we'll have a little bit of time for questions at the end.
Great, thanks, Connie. So I'm just going to take a few minutes to give a quick update on some really exciting work that was sparked by the Botanical Safety Consortium and really led to a broader collaboration with key scientists at the Malawi College of Medicine. And through various discussions, this led to a small pilot study, the title's listed there, based on evaluating traditional and complementary medicines and conventional medicines that are used concurrently in non-communicable disease management. So this gets very much part and parcel to what Dr. Lodog just talked about. The idea with this study is to look at some of the potential herb-drug interactions and or any adverse reactions that could be associated with this concurrent use of conventional and herbal medicines for patients that are being treated for non-communicable diseases. And we're looking at hypertension and diabetes, but others as well, but that's kind of the focus here. So this was discussions with our collaborators at the College of Medicine at the University of Malawi. They're listed here, and we've had various conversations with them, really great discussions over the last year or so with them, as well as other folks involved in the Botanical Safety Consortium. The concept here is to really get information and develop a survey that's going to help us look at this overlap between herbal medicine therapy and use, the population that may suffer from hypertension and diabetes, and then those that are being treated with prescribed conventional therapy. So the pilot work is a 6-month cross-sectional study, and the results are helped to inform where there could be potential herb-drug interactions that then can be further explored. So it's really just at that very, very first stage, trying to understand what types of medicines and herbs are taken concurrently. And then, of course, we are during the survey collection trying to identify if there's any adverse reactions that have been observed. Obviously, then follow-up would be needed. So over the last few months, we've been working to develop all of the paperwork, and we have a survey that's been put together, going through review within the College of Medicine for ethics review, et cetera. And we're hoping that the survey and the initiation can start in July. Things have been a bit delayed because of some challenges over the last year. Our organization, HESI, is actually providing the grant funding, and we have several BSc members, many of whom are on the phone, who are patiently waiting for me to follow up, who have been willing to serve on an ad hoc advisory group once this gets started. So we'll be in touch with those of you who volunteered to be part of this going forward. But I think it's a really exciting collaboration where we're seeing some of the expertise and interest from the Botanical Safety Consortium and HESI come together with the colleagues at the in the University in Malawi. So that's one thing that's happening. And as a result of this pilot work, as always happens, there's always interesting things that pop up. And I have to thank Stefan Gaffner for this slide, and also Larry Merritt for the picture there of the Aristolochia. During the discussions with the folks at Malawi, as well as some additional work that Stefan was looking at related to the most commonly used herbal medicines in Malawi, Stefan noticed that the Aristolochia hockii was cited as one of the most frequently used plants in Malawi. And what we know from not only some of the, obviously it's been known for quite a while, but Aristolochia fangi is one of the botanical case studies for the consortium, that we have issues related to aristolochic acid nephropathy that has been shown after use of the medicine. And the mechanism of the aristolochic acid and the end-stage renal disease is very well studied. So one of the questions, and there have been some really nice reviews, the references are down there, looking at different species of Aristolochia and whether or not they contain the aristolochic acids and in what concentrations. And pretty much what we've learned is for Aristolochia hockii, it has not been evaluated for presence of aristolochic acid as far as we know. And so one of the ideas is to develop a pilot study to analyze this species of Aristolochia, working in collaboration with HESI and other members of the Botanical Safety Consortium to try to get a little bit more information on this particular medicinal plant that is commonly used in Malawi. So this is just at the very beginning. We have quite a few discussions going on to try to figure out the next path forward for this, but it's just really exciting to see something like this come out of the discussions. And the last thing that I will mention, excuse me here, is another collaboration that really stemmed out of the discussions. And I have to thank Stefan Gaffner and Cynthia Ryder. They gave a lecture on botanical safety and a lot of the topics that the Botanical Safety Consortium is focusing on in May 2021 to students at the College of Pharmacy in Malawi via Zoom. It was really well attended, a lot of really great questions, and I think we're looking forward to continued collaborations and work with the College of Pharmacy in Malawi. So really have to thank Stefan and Cynthia for excellent lectures and hoping that we can continue to do those in the future as well. So I'll stop there, Connie, and I don't know if there's any questions people have, or if you, go ahead.
I haven't seen any come in yet.
Okay. Well, if anyone is interested in knowing more about this particular project or collaboration, please do not hesitate to send me an email and I can get you connected in, myself or Connie. Okay.
Yeah, not seen anything coming in. Yeah.
Did you want to turn it over to Dan?
Yeah, Dan, are you there?
Yes, I am. Can you hear me?
Yes.
Dan.
Right. So I would like to just spend a couple of minutes here thanking two very special people. We have two people, Doctors Jim Griffiths and Joe Devers, who are rotating off from the Botanical Safety Consortium steering team, and just want to say thank you for their leadership role in the foundation of the BSC. Dr. Joe Devers has been a critical part at several levels of the work of the BSC. As one of the original contributors to the conversation down at ICSB, the International Conference on the Science of Botanicals, he offered a lot of unique insights and technical leadership that were key to getting the consortium off the ground. Willing to help in big and little ways, Joe has been a strong contributor not only to the steering team, but as the co-chair of the Systemic Tox Working Group. And his presence will be sorely missed, I know, among those of us on the teams that he's been participating in. Just a great guy, and wish him the best. And by the same token, Dr. Jim Griffiths has also been a critical partner in the BSC. His participation also included those same conversations at ICSB, but Jim was also critically important to the inception of the BSC as he provided his international perspective, contributing his expertise to the broad-based discussions that occurred in Bratislava at Eurotox, and Jim's longstanding history with the botanical dietary supplement industry and interacting with a broad array of regulatory agencies and non-governmental technical committees. He's just been a highly valued asset to the BSC as we have sought to broaden the participation base of the BSC and just get a diverse array of folks involved, many of which are on this call today, and we are all just so deeply appreciative of the growing depth of talent that we have, and a good portion of that was due to Jim's network and his ability to get out there and beat the bushes. And so I just also would say Jim's presence will also be deeply missed. Huge thanks to both of these gentlemen as they rotate off the steering team, but we certainly hope that they will will be able to continue to enjoy their company with the activities of the BSC and elsewhere.
Thanks so much, Dan.
Yep. Thanks, guys. I don't know if either one is on. I can't tell that.
Jim's on.
Very good.
Jim, if you want to say any parting words real quick.
But I want to be clear, he's rotating off the steering committee, but he's still going to stay involved in the consortium. We're not letting him go too far. And Joe, even though he's moving to a new organization, we're hoping to loop him back in once he gets a little bit more settled. I've already warned him about that. So
great.
Okay, Michelle, maybe we can just go to that last slide. Great. And just remind everyone where our website is and how to get in touch with us, both for if you have ideas for the consortium, if you want to get involved, if you want to be on a specific work group or just have ideas, we're really open to it. And just really appreciate all the speakers today and all the attendance. We'd have a minute or two for questions if there are any. I'm not seeing any though. So I just want to take the time to thank everyone. Maybe any other comments from the panelists before we wrap up today?
Just thank you to you guys for organizing this meeting. Thanks so much. It's been great.
Thanks, Cynthia. Yeah, I think this is a good place to end then, Michelle, unless there's anything else from you.
no, I just would like to thank everybody. We do eventually really very much look forward to having an in-person meeting. But we also really do recognize that the virtual platform allows a lot more people to participate. So we'll probably, even once things open up, try to do a sort of hybrid to allow for broader participation. It's been really exciting working on this project. And I just thank everybody for taking time out of their busy schedules to join us. And as Connie has already said, I can't stress it enough, if you're not already part of the consortium and you're interested in being involved, please reach out to us, because we'd really love to get your input and feedback as we go into the next phase of our work.
Thanks, Michelle. And we hope to put the results out as they come in from the different studies with the different assays, either in the form of a stakeholder update if you're a member, or a newsletter, hopefully about quarterly to every 6 months, so to keep people involved. So thanks, everybody. I'll go ahead and end today, and just thank you so much for your time and participation. All right, bye.
Thank you.